Ladies and gentlemen, welcome to the fourth Human Cell Atlas Asia meeting. I'm Guji Guo from Zhejiang University located in Hangzhou. It is my pleasure to welcome all the participants across the world to this two-day virtual meeting. The world is now confronted by the ravages of COVID-19. The international scientific community has never flinched. We stand closer than ever. We can fight against the virus using our strongest weapons, science, knowledge, and collaboration. Today, we get together to propose and discuss about Asian contributions in the construction of the Human Cell Atlas. We will discuss about important topics such as COVID-19 research, single-cell technology, computational analysis, and genetic diversity. We put emphasis on promoting young scientists and selected many interesting talks from the submitted abstract. During the lunch break, we have recorded flash talks in the plenary meeting room. We are also eager to hear different opinions from all of you during breakout discussions. So now it is time for us to move forward to the first talk of this meeting. Let's welcome Aviv Regev, the spiritual leader of the Human Cell Atlas. She pioneers the use of single-cell genomics and other technologies to dissect molecular networks that regulate genes, define cells and tissues, and influence health and disease. Welcome.
Thank you very much, Guoji. It's a pleasure to be here virtually and in spirit. And I couldn't agree more about the fact that when we come together with science, knowledge, data, and collaboration, we really can conquer very difficult challenges. And so I'm going to give a brief introduction to the Human Cell Atlas in general, talk a little bit about where things stand in the Atlas, and also share a couple of the scientific stories from Atlas research in the context of human disease. I have to start with my disclosures. I have equity in Celsius Therapeutics and Immunitas. Until July 31st, I was a consultant for Thermo Fisher, Neogene, and Syros. And since August 1st, 2020, I'm an employee of Genentech and Roche. And so, as you know, the mission of the Human Cell Atlas is to create comprehensive reference maps of the types and properties of all human cells, which are the fundamental unit of life, as a basis for us to understand, diagnose, monitor, and treat in health and disease. And we sometimes fondly call it a periodic table of our cells. But aside from the beautiful analogy, I think one of the things that many of us hope is that, similar to the periodic table of the elements, that wasn't just a nice way to chart all the elements, it actually highlighted the organizing principles and a theory for thinking about chemistry, that studies of our cells would allow us to understand the principles underlying cellular properties in the human body. Human Cell Atlas has really kicked off in October 2016, and spent its first year mostly as a planning phase and also setting up a lot of the infrastructure and getting together as a community to articulate our vision and kick things into high gear. And since October 2017, we have been in a very active collection mode, and so we're really getting close to closing our third year for that. During this time, through many meetings, networks that have formed, and other activities, the Atlas is being collected, and at the same time, I think we all share in our scientific knowledge and expertise and really advance both technology and biological and clinical insights forward. And so a brief snapshot into the status of the HCA community. As you know, HCA is a completely open global initiative. Anyone who is willing to adhere to the values of the HCA is welcome to join. And we have just crossed the 2,000 member mark a couple of days ago. In fact, yesterday night, it was 2,003 members from 75 countries and more than 1,200 institutes, and 99 of these members are in China. That's as a little tribute to Guoji himself, across 74 institutions. And in fact, the people involved in HCA are a much larger number than that. Often within a single lab, one person from that lab would register as a member, but actually all members of the lab participate. And I urge those who are not members to join the HCA. HCA scientists are organized in different ways, and one of the ones that has really emerged now as a major principle for us are biological networks that span the different organs and systems of focus of the HCA, including organs such as, the brain or the gut or the heart, but also systems such as the immune system and particular phases of like human development. And as we will hear later during this virtual meeting, also tackling questions in genetic diversity. And scientists actually from HCA China have played really critical role in nucleating and leading many of these networks. Our overall goal is to build a comprehensive atlas over a 10-year period, and we are well on our way for our first draft of about 100 million cells. And just like this graphical rendition, even though we have partial information as we build the atlas, even the partial information is incredibly useful for us. So just as a quick snapshot of where we stand across about 12 of our systems, in many of these systems, here are the liver, the kidney, the skin, the lung, the gut, and human development, we have substantially passed the 1 million cell mark, and in almost all cases, the 500,000 cell mark. And we have also analyzed samples from dozens and sometimes hundreds of individuals. That actually indicates to us that we're getting from a census perspective, just knowing the cells that are there, to a point that's going to get close to saturation for many of the organs, and that can assure us that we're already recognizing also very rare cells. At the same time, this community has really sprung into action around COVID-19, and in many of these organs that are actually highly relevant to COVID-19 patients, such as the lungs, the kidney, and the liver, we also have cells now that have been profiled from COVID-19 patients, and I will come back to that. To show you 6 of the additional organs, in particular in the brain, there is a fantastic effort that is led especially because of the Brain Initiative and has crossed the 10 million cell mark, but also in systems that in the beginning of HCA were not at the forefront, such as reproductive systems in the that are really giving us a very substantial glimpse and level of information. And finally, in the immune system, which is one of the more accessible systems for us for single cell analysis, we're well on our way to the 7 million cell mark. And again, in the relevant tissue, also information from COVID-19 patients, especially in PBMCs. And I believe that Sarah, who is going to talk later in the symposium, is going to cover that aspect. I also wanted to give a glimpse into part of our mission, which is to use the reference atlas as a context by which to understand not just health, but also disease. And the two are really inextricably coupled to each other. If we know what health looks like, we understand disease better. But also by looking at disease, we actually understand what homeostasis and health looks like as well. And in the context of a disease of a disease study, there's a series of questions that scientists are interested in, like where might disease risk genes from human genetics act, which cells might be disrupted, how are their cell programs changed, what happens to their functions, how are the cell communications disrupted, what might be the effect of drug. And it turns out that a healthy reference atlas can be very useful for many of these queries. So I'm just going to give you a glimpse into one particular query, which is maybe the simplest one that you would ask of an atlas, which is, where is my gene? And in that context, the Human Cell Atlas actually has developed a really close partnership with human disease atlases, both globally and through specific initiatives. And so if there is particular initiatives that support healthy reference studies, they work now really hand in hand with human disease studies. And I want to highlight in particular the Lifetime Initiative from Europe that has really taken a strong disease focus complementary to the HCA. And so in the two examples that I'm going to show you, I'm going to talk about common disease and how we can do queries for genes to really help us understand better genes from genome-wide association studies. And then I'm going to turn and talk a little bit about COVID-19. And so for my first example, I'm taking the context of the gut cell atlas and how we might try to use it in order to understand a complex genetic disease like ulcerative colitis, which has really been a poster child of human genetics because it has allowed us to identify many new causes and in many cases highlight new pathways that we didn't ever suspect would be involved in ulcerative colitis. But for many of them, we do not know the cell of action, and we do not also know the process in which the genes participate. And so amongst the cells that I've shown you for the gut, which have now well past the 1 million cell mark, about 265,000 of those cells were collected in a study that looked both at healthy tissue and at samples from ulcerative colitis. And in this context, we can develop an atlas, and this atlas combines both the healthy and the disease cells together, embeds them together, and says 51 different subsets of cells, in this case, only in the colon mucosa, which is one layer of the colon from both healthy individuals and 17 individuals with ulcerative colitis. Doing this and having a healthy reference allows us to ask the most simple question about disease. Has something happened to its cells? And we find that there is a dramatic remodeling of the gut cell composition. And we can only do that because we can compare the disease tissue to the healthy reference atlas. And in this case, we might find that there's M cells that are present only in ulcerative colitis and typically are not actually prominent in the colon at all. There's these fibroblasts that assume this inflammatory state, which we again do not see in healthy colon. And then there are these T cells that express both CD8 and IL-17 that are greatly expanded in ulcerative colitis. Now, if we turn to the genes from genome-wide association studies, given that we have this beautiful catalog of 51 subsets of cells, we can ask very easily, where might they be expressed? And so we're focused here on those genes that are actually, where the genetic signal was already narrowed down to a single gene level. And we look at them in the columns and we consider their expression across all 51 cells in this colon mucosa atlas. We can see that we can identify cell type specific expression both in healthy and ulcerative colitis, irrespective of the condition. There's genes that are expressed specifically only in healthy tissues, and there are genes that are expressed specifically only in ulcerative colitis. Now we can go to a particular cell type, such as these M-like cells that are expanded in the disease, and we can look at their enrichment for genes that come from GWAS. And we see that there is a preponderance of genes from genome-wide association studies that are actually expressed in M cells, and moreover, those genes are induced in those cells under inflamed conditions. So this is a very simple but effective use of an atlas, but there's something that I didn't tell you. And so I showed you the genes that have cell type specific expression. There are about 50% of the genes that were already mapped to a single gene level in the locus. But what about the other 50%? Now I'm showing them as well. They're to the right of the plot. And you can see that those genes are either not very specifically expressed or honestly not that much expressed at all. And so there could be many reasons for this, including interesting biological ones. But the most prominent one is that we didn't actually profile the whole colon in this initial atlas. It was only the mucosa layer, which is only one layer. There are cell types like adipocytes, for example, that are only present in deeper layers like the muscularis layer. In addition, there are some cell types that are still missing from this atlas, and in particular, very rare cell types like enteric neurons, which are not just rare and so harder to capture at large numbers, but they actually do not survive dissociation of the tissue into single cell analysis. And so we have recently developed 2 lab protocols. One is called RAISIN-seq that allows us to do better single nucleus RNA-seq, which allows us to capture neurons, and the other called MIRACLE-seq for label-free rare cell enrichment in order to get a more comprehensive view of the tissue. And when we do this and apply it in this case to frozen resections, healthy portions of the colon, we obtain another 150,000 profiles, amongst which are, for example, 1,500 enteric neurons, but also other rare cells like interstitial cells of Cajal and adipocytes. And when we do this and we go back to the same plot that we looked at before, it now turns out that many of those genes that were not expressed at all or expressed lowly or not specifically in the mucosa have very specific expression patterns inside these additional layers. They might be expressed in adipocytes, they might be expressed in subsets of enteric neurons. And moreover, now we can start and look at other types of diseases that might not affect the colon mucosa, but for example, where we expect to see an effect in the enteric nervous system because there is a gas dysmotility phenotype, such as a rare disease like Hirschsprung's disease, or common diseases that are typically central nervous system diseases like autism spectrum disorder and Parkinson's disease, but that have a gut dysmotility comorbidity. And so this really highlights to us the importance of systematic characterization of atlases. It's not enough to have a portion of the human body because genes are everywhere and you shouldn't assume that they're necessarily where you expect them to be. So here I really want to give a shout out to Guoji. This is his human cell landscape rendered on something that looks a little bit like the globe. So we thought that was cute. But it's really one of the first examples that we have of a cross-tissue atlas, where you can start asking these questions without assuming that you're necessarily in the right tissue, because maybe the gene is actually important in an additional cell type in another tissue. And most recently, we have deepened this kind of approach, in this case, in the context of an autopsy study looking at 8 different tissues and examining, in the end, 37 different cell types across these tissue subsets, seeing both substantial commonalities between tissues as well as a lot of tissue-specific expression, even in shared cells such as immune cells, stromal cells, and so on. Most recently, we have started taking this approach one step further, trying to combine the single cell signal together with the human genetic signal in an analysis of heritability to try to say which cell carries sufficient heritability signal for particular diseases. We developed this approach together with Alkas Price, who's a human geneticist, and now we can look at the heritability for inflammatory bowel disease, a signal coming from human genetics, and cross it against the atlas, first looking at the healthy cells and their cell type specific expression. We see, for example, myeloid cells, which is well appreciated for this disease, but for example, also these M cells that before were not known to contribute to disease risk or barely were known to even be there in the colon. You can also do it now with gene programs that are specific to the disease conditions, such as in ulcerative colitis, and now we find the fibroblast-associated disease program that carries heritability for the disease. And finally, you can do this systematically for each of the cell types, asking both on the cell type and on the disease condition, and you see that enterocytes are important in IBD overall, and especially in ulcerative colitis, whereas ILCs are important in IBD and Crohn's, but not so much in ulcerative colitis, trans-amplifying cells, the opposite, and T cells across the board. And so the second quick vignette that I would like to give you has to do with how we use the atlas to understand a disease when it's emerging, when we don't have, hundreds and thousands of papers in the literature as we have for inflammatory bowel disease, but we're all living through it in our own backyard and in our own neighborhoods and communities. And of course, this is COVID-19. It's a beautiful story also of our community coming together. And so there's many different questions that were and still are open in the context of COVID-19, including transmission. Sarah will speak to that in her talk later on, the epidemiology of the disease, why we see it hitting some subsets of the population more than others, the pathogenesis, the disease mechanisms, and also the viability of model organisms, which we need in order, for example, to develop therapeutics. And I like the example of human genetic studies in the context of the Human Cell Atlas. The first question that one would want to ask is, where are the cells that the virus might infect? And here, of course, by February, it was already quite well established that ACE2, which is the receptor for SARS-CoV-2, along with it needs an accessory protease, TMPRSS2 and cathepsin L, and possibly others could be those receptors, and we can use those genes as a very simple query across the atlas. I won't belabor this. This is published work, but or posted work on the bioRxiv. But the critical piece for me to emphasize is that this was an international effort across the entire Human Cell Atlas community. It really started with the lung biological network because our original focus was, of course, the lung and the airways. But as our understanding of the disease evolved, as well as just our curiosity of the possibilities increased, then actually people from the heart network and from the liver network and from the kidney network and so on have all joined in. From the initial study, we have just from the healthy atlas, we have learned a lot. We have learned about cells involved in transmission, about the potential for transmission through the placenta and the fetal lung, so vertical transmission, the different tissues in which pathogenesis might occur, which actually track extremely well, unfortunately, with the pathology that we see in patients, in the involvement of pre-wired immune-like programs that are in the cells that are likely infected, different epidemiological aspects, including the association with age, sex, and smoking status, as well as the suitability of model organisms, both the mouse and non-human primates. But as time moved and as we all are living through this experience, then our same community really rallied around, in the spirit of science, knowledge, data, and collaboration, our community has really rallied around studying with single cell technologies and spatial technologies patient samples. There are studies of the blood, of other specimens that you can obtain from living patients, such as aspirates, nasal swabs, and lavages. Sarah will speak to those. And in addition, there are multiple efforts focused on autopsies in order to really access tissues in the depth of the body where the virus is really wreaking havoc. And I will focus on an autopsy study like that for the next 5 minutes or so. And so there are several such efforts. I'm highlighting two on this slide, one coming from the Boston area, and I'll focus on that one. The other coming from Imperial College London. There are also efforts in New York, multiples, in Arizona, and in multiple locations in Europe. In a typical study, such as this one, this would be considered a relatively large one. These are, of course, very difficult studies to conduct. This one has studied 17 different patients across a broad range of ages, different types of organ failure that lead to death, from lung to multi-organ failure, in one case neural complications, and a collection at least of lung, heart, liver, and kidney in almost all patients, and then from a subset of the donors, different subsets of the tissues. And for those who live in the United States, the health disparities and the high impact on African Americans and Latinx individuals is really emphasized in this table. What do we do in a study like this? Well, we apply our Atlas toolbox. All that we have learned to do in other settings, we can apply to COVID-19. We can obtain fresh tissue and try to process it to single cells. We can obtain frozen tissue and process it to single nuclei. And we can obtain fixed tissues in FFPEs and perform spatial analysis. And we've actually done all of those things. So first of all, to emphasize the great challenge and difficulties for scientists to do this. So these are three heroes who worked in the BL3. Two of them have never worked in a BL3 before this pandemic, but they were experts in single cell and single nucleus RNA-seq. And the third was a BL3 expert, but has never done single cell studies. And these types of studies start in the autopsy suite, in the hospital, and through a very rapid style of communication and little kits that you invent on the fly, they end up in a BL3 facility for processing. You also need to set up the right processing timelines. These are pipelines. These are far from perfect studies. So for example, we have to, these autopsies sometimes are conducted more than 24 hours after death. And so there's plenty of ambient RNA in the tissue. We partnered with the cell vendor developers in order to remove this ambient RNA. You have to annotate almost a whole human cell atlas cross-tissue study in a very short amount of time. So we developed a transfer learning approach for that. And you also want to study where the virus is, so you have to have a system to analyze viral RNA at the same time. And without belaboring all the technical steps, this is an example of the lung portion of the study, 70 lung autopsy samples, almost 37,000 cells. You can see the nice overlap between the patients. You can see the annotations, and you can see both some repeatability as well as quite a bit of variation in the composition of the different samples. Now, of course, you can go back to the context of the healthy reference, and just like the UC example that I showed you, you can ask, how does this compare to healthy tissue? And also because Atlas studies, such as in Naftali Kaminsky's lab, are also looking at other lung diseases, such as in this case, idiopathic pulmonary fibrosis, you can compare also to other types of diseases. And we see that across the three major compartments, stroma, immune, and epithelial, the COVID-19 lung looks different from both IPF and healthy lung, but also the same is true also when you dig into finer annotations and the compositions of the cells. You can also start asking questions in the context of human genetics. So in parallel to this effort, there is an international effort to conduct GWAS of COVID-19 severity. Several of the loci have already been mapped, for example, chromosome 3 locus and chromosome 21 locus. We can now look at the genes in these loci and try to help the human geneticists in the same way that has been done in UC in order to know in which cells are those genes expressed in a relevant tissue, such as the COVID lung, as well as in the healthy lung. Now we can start digging in, and we can ask about expression changes in each subset of cells. So for example, 82 cells, which are a major target of the virus, you can ask, what is the difference in expression profiles of 82 cells in COVID-19 lung versus healthy lung? And we find reduced expression of surfactants and reduced anti-apoptotic activity, both of which are consistent with the phenotypes we see in patients, and an induction of inflammatory, innate immune damage response, cell migration, and ECM pathways in the cells. Along doing such studies, you can actually reach some meaningful scientific insights. So for example, one of the things that we found is this enrichment of a gene program known as PATS for pre-alveolar type 1 transitional cell state, which is a gene program that gets activated when 81 cells, which are responsible for oxygen exchange, when they're damaged and there isn't enough stem cell capacity to replenish them, 82 cells can transdifferentiate to 81 cells. They do it through a transitional state, and sometimes they can get stuck in this state. This can happen in fibrosis, and it seems to happen in COVID lung as well. Another aspect of this is a change in the fibrotic, in the fibroblast compartment, and we're still looking into that. Finally, you can ask, where is the virus? And so for this, we use viral reads. We take our annotated atlas, we look at the entry factors, and we actually find that the viral reads, by the point of time that we're looking at, which is when people are unfortunately deceased, the viral reads are not actually in the cells that have the preponderance of the entry factors. And this can be for various reasons. For this, you need to know in which cells they are. So we can relate viral RNA reads to ask about whether viral RNA reads are related to cell infection. We ask about alignment to the minus strand versus the plus strand. We have some indication that it might be related to it, but we cannot definitively say that this is productive infection. There is dramatic variation in the detected viral RNA in the cells across patients, and we find that macrophages and vascular endothelial cells have the highest number of cells with viral RNA. And as we drill into these two subsets of cells, in macrophages and endothelial cells, and compare cells with and without viral RNA, we see that they express different gene programs, which are actually consistent with a response, at least to viral RNA, not necessarily to viral infection. Such as, chemokine-mediated signaling, type 1 interferon signaling pathways, and so on. So finally, for the COVID-19 story, I want to turn very quickly to looking at it also with a spatial perspective, because of course our atlas studies always have both a cellular and nuclear arm and a spatial arm. And so first, just to give you a glimpse of what a COVID lung might look like, in pink is actually SARS-CoV-2 probe. This is with RNAscope, and you can see the very high level of infection in this particular lung, but you can also see that it can be very specific to particular parts and doesn't actually go into other adjacent parts. So in order to get a nuanced view of the spatial microenvironment, and since we had to do this work actually with fixed tissue in FFPEs, we turned to the nanostring GeoMX DSP, which allows us to first identify a region of interest and then specifically capture the RNA from an area of interest within that region of interest. This is an example from patient SO3, and on the right, at the bottom, you can see the mask that we apply in segmentation and capture only the RNA from the mask, from the region of interest. When we do this, we can take then each of the profiles that we obtained from each segment, an epithelial segment on the left, a non-epithelial mass region on the right. We can compare we compare here 3 healthy lungs to 7 COVID-19 patient samples. And we can take the profile from that region together with the cellular profiles we have from the single nucleus analysis and infer the cell type composition for each region of interest. And by this, we find that there is a reduction in mast cells and an increase in macrophages and myofibroblasts in COVID-19 versus control samples. Now we can go within, compare patients and controls, and we can compare the COVID-19 high patients to the controls and identify the pathways that are activated in the alveoli in both epithelial and non-epithelial cells, and find different types of both interferon alpha and interferon gamma and other immune responses. We can also ask within the same patient to compare COVID high versus low regions of interest, which are both epithelial, and identify a unique response to the infected cells that we don't see in the non-COVID infected cells, which are epithelial and in the same patient. And those involve, again, interferon alpha and interferon gamma response and other antiviral responses. Coming up, liver, kidney, heart, and additional atlases from the same patients, which would also allow us to compare within the same patient for different things. And as I said, Sarah will touch on other aspects of this major effort in the community. We have set up multiple resources, including in partnership with the Lifetime Initiative, to allow the community to register their interest in this kind of research and to find partners, experimental, computational, clinical, to understand the data that we're all collecting. In the last couple of minutes, I want to turn back to why we're able as a community for HCA to pivot so quickly. And this is actually built on all of the work that we've done and are doing in the Human Cell Atlas Initiative over the past several years. We were ready to go because we were collaborating already, because we had infrastructure, platforms, methodology, and an extremely strong sense of openness and community, starting with our values. And within them, I want to really emphasize community, equity, our transparency and open data sharing, and our commitment at the same time as we study human samples for privacy and ethics. These things that really permeate through our working groups. So in our analysis working group, led by Dana Peer and John Marioni, in addition to organizing data analysis teams, meetings, and jamborees, some of the methods, such as the ambient cleaning method, is an offshoot of a jamboree that happened quite a while back. But also recommending experimental designs and analysis frameworks. The data coordination platform at data.humancellatlas.org, where we leverage a data store that has built-in support for managed access, which is coming up soon, but not just there yet, and that will have a data access committee to facilitate access to controlled access data, which would really improve our ability to do human studies. And the cell annotation platform, I already highlighted the annotation as both a major challenge and one of our greatest opportunities to come together quickly, which is led by Peter Karchenko, working together with Fabian Theis and John Marioni. The lab protocols that I highlighted, such as all of those nuclei protocols, are really embraced by the efforts of the Standards and Technology Working Group, the STWG, led by Holger Hein, Orit Rosenblatt-Rosen, and Zhouwei Zhuang, spanning both single-cell and spatial methodologies, and having gone through multiple cross-community comparisons of protocols, yielding more and more papers along the way that really allow all of us to move better and faster. Our equity working group, our most recent working group, but an activity that actually started in 2016, led by Musa Mlanga, Partha Majumdar, and Alex Shalek, is really committed to promoting and supporting progress towards equity in the HCA. In the COVID-19 context, this played a critical role in the ability to really compare to other infectious disease, because this is something that the equity working group has been committed to as part of its global efforts. And it works not just to ensure that our atlas is diverse in its science, that our samples are diverse, and I believe Jay will speak to that, but also that it is diverse in its scientists and in where they are in the world. This has to be a truly global effort. In June, in the context of both local events in the United States and more globally, we came up with a strong statement on our commitment not just to Black Lives Matter, but also to global ethnic equity. And we identified specific actions, including our current commitments to expand outreach programs in underrepresented countries, especially LMICs, to implement regular diversity audits for our membership, our leadership, our study participants, and our collected data, and address the gaps, and to work with funders to encourage the earmarking of funds to increase recruitment of scientists and study participants for underrepresented regions and communities. And I'm thrilled to say that the CZI has actually very recently announced a supplement that it would provide its grantees in order to meet this specific action. In addition, one day when we travel again, we will have travel stipends. In fact, the virtual world has improved our equity standing, and it's something to really appreciate. And we're in the process of running a survey for equity, as well as have done at least one equity roundtable to brainstorm additional solutions. And finally, our ethics working group, led by Bartha Knoppers, Barbara Wald, and Roderick Luijo, and really facilitated by Emily Kirby at McGill, working with Bartha, allows us to make sure that what we do really serves humans. It doesn't just serve our science, it serves the patients, it serves the individuals, it serves the people who so generously donate and agree to participate as research subjects, without whom there would be no human cell atlas. And so they focus on an ethics toolkit, developing launching our ethics help desk, and ensuring that people from across the world can really support. And so I hope I gave you a glimpse today of where the Human Cell Atlas stands, of our commitment to bring together the community and our science. No time like the present reminds us how important it is to have data-driven approaches to biology. Mysteries still abound, and how important it is to be global, open, and collaborative. We're excited to see the steps ahead as the Atlas grows and becomes more and more complete. And with this, I will finish and be happy to take any questions that Guoji sends my way.
Okay, fantastic talk. Can everyone use the Slido to type in your questions? So we have a question from Zeming now, and he's asking, hi, Aviv, in your COVID-19 studies, did you detect viral sequences in non-epithelial cells? What are they? How real are those? And do those cells with viral sequences mean direct infection?
This is an excellent question. I tried to touch on it, but I really was a little short on time. First of all, they're real. They are real, they are significant, they are not ambient RNA. That is one of the things that we have devoted a substantial amount of effort for, because we wanted to be certain. And we also know that when we bulk them up, we actually get the result of bulk viral sequencing. So we have another additional control that I didn't even touch on. Their preponderance in macrophages needs to be taken carefully, not with respect to their realness. They are absolutely real, but they can be the result of phagocytosis. And it is very difficult to rule that possibility out, because the one tool that we have in our arsenal using sequence to try and distinguish things, you can look at basically some version, right, of transcripts that you would expect to be present only in a replicating virus. And here is the difference between the minus strand and the plus strand for SARS-CoV-2, and others have done it with splicing. When you do that, you do see some signal that is indicating of a virus, I would say, in the process of being replicating. It is a much lower signal, which is to be expected, but it is substantially lower. And moreover, it still cannot rule out in a macrophage the possibility that it phagocytosed a cell that was not a macrophage, in which there was a virus that was replicating. So we are extremely cautious about concluding that this is a signal of replication. There are in vitro studies from other groups that have shown that as you progress in the very early stages, there is a signal like this in the minus versus plus strand, but we can't extrapolate it. And especially because these are such late stage, I mean, these are autopsy samples, these individuals have passed away, we're surely not seeing the first wave of viral infection. In the endothelial cells, which is the other major cell type in vascular cells, vascular endothelial cells, which are the next major cell types in which we see reads, it's a similar story. And we did not feel the confidence to say that this is productive infection. Having said that, it is absolutely where the reads are. It's not an artifact.
Okay, so I have a question actually. I'm still, continuous, always asking myself. So in the Human Genome Projects, people do sequencing, and every base is a base. But for our, Human Cell Atlas project, and there can be lots of variations in digestion and data quality. So how, what we can do to improve those, the integration of the ultimate atlas?
So that's a great question. First of all, I want to say something about the human genome and base errors. Every base is a base in reality. Sequencing makes a lot of errors. We all know that. People knew it then. Coming up with quality metrics was not just it wasn't an obsession. It was absolutely justified. It took years and years and years. It's not that when the Human Genome Project started, everyone got perfect base calls, and errors existed and errors subsisted. And depending on the sequence that you were sequencing, there were regions that took many, many years to finish after we had a draft human genome in 2000, and there's regions that are not finished to this day. There are still gaps. So first of all, there's the completeness and the error question, even for the human genome. In the human genome, you have a second layer, which again, I think is really timely for this meeting, which is the aspect of human genetic variation. The Human Genome Project didn't answer the question of human genetic variation. It provided the scaffold on which human genetic studies could act. So similarly, I would say for us, I think we could actually go a little faster and a little better than we originally anticipated, because we got the technologies better and they got improved with time. The question you ask, though, is a different one, because it's a question that asks about the variation between cells, which is driven from technical aspects versus the variation that is driven from non-technical aspects. One of the things that is very comforting, or increasingly comforting, at looking at these studies that now span dozens of labs, hundreds of samples, millions of cells, is that in fact, when you do all these lung studies, they kind of agree with each other. They're not radically different from each other, even though there are differences. There are differences between doing nuclei and cells. There's differences, of course, between the regions that you actually study in the lung. But one thing that the COVID-19 effort made us do more rapidly than we might have done otherwise is that we got this huge view of more than 20 something studies by now of the lung. And when you look at all of these 21 studies of the lung, and they weren't even processed together, not just experimentally, they were processed computationally by their own researchers. There's a lot of limitations on what was on what could be done in such a short time. When you look at them, at the end of the day, once you control for the fact that not all of these studies sampled the same region, studies of the same region with the same strategy gave pretty much the same results. So I would say, you know, just like human genomes, there's variation, but by and large, it's pretty resilient. Our computational techniques got a lot better. Our experimental techniques got better too. So by now we're in a pretty solid space. And one of the benefits actually of the way that the atlas is being constructed by multiple groups doing the same organ in parallel but coordinating through a network is that we have a real sense of this variance. Because if everything would have been done in one place, maybe everything would have looked really clean and tight, but some of it would actually be systematic error, rather than it would be the error unique to that. And that's actually a lesson learned from sequencing studies, that you actually don't want to do them all in one place because each place has its own set of errors.
That's a great answer. Thanks. So because of time, we have to move to our next speaker, Jae-Shin. Jae-Shin is a team leader from Riken Center for Integrated Medical Sciences. He's co-leading international consortiums such as Phantom 6 and Human Cell Atlas. Jay enjoys building functional genomics platforms to decipher the molecular mechanisms involved in gene regulation, cellular plasticity, and disease.
Okay, good morning, everyone, and good evening, good afternoon from other parts of the world. Thank you, Goji, for introducing and also for putting this wonderful virtual meeting together. And it's a great pleasure to introduce to you about the Human Cell Atlas Asia efforts. And as you have heard this fantastic talk from Aviv just now, there has been a tremendous effort globally to build this Human Cell Atlas. So the question is, what can the Asian community do, and how can we forge a collaboration that can also not only represent, but also lead in areas that can really contribute to the global issues such as disease and COVID-19. So today I'm going to kind of give you a past history of what we have been doing and what we hope to do in the future, and hopefully that this can rally up some of the community members in Asia to communicate and to collaborate and to forge new research ideas. So let me see if I can change this slide here. Okay, so as you can see, as you have seen also from Aviv, there's a fundamental mission that we all abide by and really want to make sure that we can create this comprehensive reference map across the human genome. Across the human body. And one of the ways to do this, of course, is through collection of multiple cell types from multiple locations. And one of the key ideas that really stood out to me when I first joined the Human Cell Atlas is that there are key principles that we really want to abide by, and that really is the community and diversity aspect of the Human Cell Atlas. And this quotation is just straight from the HCA white paper, which is that, you know, cells will be studied using broad range of techniques to capture both breadth and depth that will fully represent the world's diversity. But furthermore, the Atlas is, the HCA community is committed to genetic and geographic diversity and equity at every phase. So inspired by these missions, we really wanted to make sure that we can also come together as scientific communities in Asia to represent and fulfill such missions to cover and to fulfill the completeness of the Human Cell Atlas. So I'm kind of going to introduce to you, many of you are familiar as we are in Asia, but I think this is a good reminder that there's a lot of interesting, abundant genetic, ethnic, cultural, even environmental and social economic diversity in Asia, because we are home to about 60% of the world's population, where the two most populous countries like China and India are in Asia. I think in contrast to Europe or the United States, there's a bit of a challenge that there's lack of central government that really incentivizes the scientists in the region to come together to collaborate on. And that's really kind of one of the key missions of the HCA Asia, is to break down barriers and being able to find common interests to bring research that can benefit the societal needs of the region. And this is kind of represented in this graph here, in that if you look at the global genomic data, that only 1 or 2% of the genome is of Asian ancestry. And this really limits the clinical utility of how we can use genomic data for Asian population. And this is another study from GTEx, where, of course, it's a US study, but only 1.3% or so is representative of Asian descent. So this naturally, invariably contributes to the biases in clinical data, genomic data, which will have a negative or unexpected outcomes for clinical implications. So with that backdrop, in 2017, Piero Caninci and myself actually organized a first Human Cell Atlas Asia meeting in Okinawa, Japan. And there the purpose was very clear: to meet, discuss about what are the latest science and technologies of single-cell genomics that are happening in the region, and to identify what are some of the societal needs and challenges and how we can overcome them, but also to strategize increased participation, representation, and leadership in Asia. And as you can see in the picture in the bottom left, it was a rather small group. It was about 40 members or so, but it was very intense, focused discussion that have led to very good network of collaborations that in 2018, Woon-Young Park and Sung-Young Kim decided to organize the second HCA Asia meeting in Jeju, South Korea. And just last year in November, Shayan Pravakar organized a third HCA Asia meeting, which had about 300 participants to really discuss these issues of how to bring science and technology in the region together. So I just want to give you some takeaways from these meetings and how this have led to our current standing and how it's going to, we hope to see pan forward. So we noticed from all of these discussions in these meetings that there are very interesting and active single-cell research that are happening with many shared interests. And I just want to kind of give you some of the highlights of the research that are currently ongoing in various countries in Asia, going by different countries. But hopefully by end of these highlights that we can find areas that could come together. So for example, in Japan, there's a lot of efforts in technology developments, such as Ramda-seq or single-cell cage-seq, but also 3D imaging called Cubic that has been very powerful to build a atlas based on tissue clearing technologies. There are unique samples also in Japan, such as supercentenarians, which will be presented by Piero later in the symposium, but also IPS technologies and how it can be used for regenerative medicine, and creating a correct cell annotation for that is one of the key programs that is really important in the country. But also later in the symposium, you'll also hear talks about how we can use organoids to study COVID-19 research. In South Korea, they've been active in Human Cell Atlas from the beginning. And they, as I mentioned earlier, they hosted our second HCA Asia meeting in 2018. And because of their dedication and support for the Human Cell Atlas, they were actually able to formulate their own group within South Korea, which they have their own steering committee, and was able to reach out to their local governments to acquire single cell program funding, which led to six projects involving proteomics, spatial, longitudinal studies, and cancer and immune disease. That also extends to colorectal cancer, lung cancer. And I think recently, Unyang Park and others are further exploring the research of COVID-19 patients using single-cell technologies. I think Singapore has also been very active in single-cell research, and I will not go through every single one of them, but their involvement in cancer has been very impactful, as well as the immune systems, studying the inflammatory dendritic cells, as well as brain macrophage development. But also computationally building algorithms have been very big impact from Singapore. I think it is also important to highlight that the hosts of this meeting, the China, have also had tremendous efforts and contributions in single cell research. I think in 2019, the scientists in China has gathered to kick off this primate cell decoding project called PCDP, and it's currently led by Sunyi Zi, Wenzi Ji, Zhu Zhang, and Xiaokun Wang. And all of these efforts are really to decode the genetic regulation in primate cells using single cell technologies. They've also come together to build atlas of blood cells, and that's led by Tao Cheng and others. And it's really to aim to build a reference map for all hematopoietic cells. And I think it is selected as top 10 medical advances in China in 2018. Again, as we all know, Go has been pioneering this cell landscape projects, and is aiming to construct cell landscape for major species. And this really shows how genome encodes a cell landscape using artificial intelligence. And so far, they've created more than 3 million cells that have profiled across human, mouse, zebrafish, and Drosophila, and others. I think China has also been leading in the front of technology development, and I think there are many ways to utilize and contribute to the global Human Cell Atlas as well as HCA Asia efforts. So I just wanted to kind of summarize that, yes, there are active single cell research that are ongoing with many shared interests, such as immunology and cancer, and that there are unique technologies and methods that can really be broadly distributed and shared for highly standardized and harmonized approaches. And I think it's also important to note that in Asia, there's tremendous genetic and environmental diversity, that we have access to unique disease cohorts, which again, in return, have high clinical utility of the data set. But I think we could also be aware of the challenges that we are facing in the region. And that is that there are varying local regulations, such as ethics and data release policies. And how we need to overcome this requires a constant dialogue, not only among scientists, but also with policymakers and also the public awareness. And the second challenge is that many funders, especially in Japan and others, are shifting towards more translational disease studies rather than basic science or technology development. So we hope that we need to make sure some of our research are meeting the needs of the funders, as well as advancing in basic research. The third challenge is that we do face in Asia is that 29 out of 49 countries in Asia are considered emerging and developing countries. So this will touch upon the issue of equity and being able to reach out to these nations to support and train the local scientists in order to contribute their scientific knowledge into the HCA efforts. And the fourth challenge that we recognize is, as I mentioned earlier, the lack of central government or the funding body really makes it challenging to incentivize scientists to build communities. And in return, we also have decentralized tissue or biobanking platforms, which also makes the data standardization or sample coordination a little bit more challenging. So whenever there are challenges, we believe there are opportunities. So we believe we need to, it's an opportunity for HCA Asia community members to come together to build harmonization of data and standardization of the workflows, and really to identify common interest projects that can benefit not only a single country, but the whole region and the population in the region. So one of the ways to do that, of course, is through organization of these meetings, to have discussions and breakout sessions to talk about these common themes and research ideas. And in the meantime, provide opportunities to promote equity, gender balance, and ethics awareness to the region, so that some of the data and scientists are equally represented. And hopefully through this, we can create opportunities to co-apply or support local and international fundings. So just to go over this quickly, to facilitate a lot of these efforts, we are in the process of creating a HCA Asia steering committee. And really the value of that committee and the main mission of that committee is to build a community in Asia to enhance the communication with local scientists. And that we can also use opportunities to showcase regional science and strengthen regional research alliance, and also to identify synergies, and in the process, diversify the Human Cell Atlas program. So the committee will constantly work with the global HCA to coordinate these annual meetings and to collaborate with also equity working groups to provide the right communication aids and ethical toolkits. And we will build communities through identifying key research themes that can benefit the region. So here are just 5 examples, such as cancer, diabetes, infectious disease, aging, and genetic diversity. We really hope to rally the scientists in the region to touch upon these research topics to expand and collaborate. And hopefully we can use that opportunity to co-apply for both local and international funding, and that the data will be contributed to the Human Cell Atlas. And I believe that all of these efforts will help us to achieve this equity and diversity and reach the high clinical benefits of the genomics data and meet the local societal needs. So I'm just going to go through really quickly these 5 flagship projects because from tomorrow, actually there's a breakout session, and we will really hope to engage with the participants of this meeting to discuss how we can build communities around these 5 themes. So one of them is called cancer, and I believe cancer is a global issue, but there are very specific concerns in Asian region that speaks to the local communities. And here's just an example of a colon and rectum cancer that are highly prevalent in Japan or Singapore, but conversely, we have uterine and cervix cancer, which are more prevalent in Cambodia and India. So just recognizing and identifying these concerns and issues that are prevalent in different parts of Asia, and being able to provide the right reference data set for the healthy, but also providing the right toolkits to generate single cell data will be critical. I think diabetes is one of the key important issues in Asia. I think there's a rapid growth of diabetic cases in Asia compared to any other ethnic backgrounds. And that could be partly due to changes in diet and lifestyle. However, there are key indications of genetic contributions that may have contributed to diabetes onsets, as well as resistance to certain diabetic treatments. Here in the Department of Health and Human Services in the US, they state that about 40% of Asian Americans are more likely to be diagnosed with diabetes the non-Hispanic ones. So again, even in the same environmental factors, there's a higher risk of Asians for diabetes, and something that you can really come together as a community in Asia to tackle this issue. Third is infectious disease, and we all recognize this through recent times, but also there are local disease burdens and local pathogen strains that just differ from many parts of the world. And due to the lack of central governance in Asia, it really makes it challenging to standardize a lot of the efforts to address some of these infectious disease. So hopefully we'll have an opportunity to talk about what are the areas and hurdles that we can overcome to address this. Aging is, I say here, is a disease of the present and future. As you can see, the highest ranking cause of disease is Alzheimer's disease in Japan and South Korea, or age-related neurodegeneration, compared to 2007, where it wasn't the case. So there's a growing number of aging population in developed countries, and we envisage that in the future, this will most likely be the case in many other countries in the world, including in Asia. So preemptively addressing some of these aging questions would also help the communities to come together and tackle some of these challenges. The fifth item that we would love to discuss tomorrow is genetic variation, as a work by the Genome Asia 100K Consortium. And there are about 10 different ancestral groups and lineages in Asia, which have a tremendous impact on the human disease and pathology. Therefore, just understanding the genetic basis across the population in Asia were critical in diagnosis and prognosis of disease, and even the treatments of particular drugs for the treatment of disease. So understanding the genetic variation as a backdrop in the context of disease will be extremely critical. And to partly address some of these issues, some of us in the Human Cell Atlas Asia Committee have initiated a group called Asian Immune Diversity Atlas. And this is a project, a pilot project that are currently funded by Chan Zuckerberg Initiative. And Chung Cha-Hon and Kawakami-san and Jay Shin, myself at Weekend, is working really closely with Eun-Young Park in Samsung, Korea, and Shayan Prawakar in A-Star, Singapore. And the goal really is to profile the human peripheral blood mononuclear cells across multiple individuals. Here, we're aiming by 500 individuals, and using multiplexing strategy that we will hope to understand how the genetic variations attributes to gene expression patterns across the population in Asia. So we have built up a very strong network of community, and we have a Slack workspace, we have Google Drive to exchange files, and we are successfully multiplexing 16 individuals into single 10x runs and generating a very cost-effective profile of the local population and the immune cells. Here is just a snapshot of integration of the human PBMCs from 3 different sites, Japan, Korea, Singapore, that we're successfully integrating them. And we are also profiling their genotype in order to understand genetic variations and how that attributes to gene expression. So I just want to acknowledge CZI again, and thanks to their efforts, we are now expanding AIDA, which will include Thailand as well as India. And hopefully we can further expand these efforts across Asia, including China and Vietnam and other parts around the world. So if you are ever interested in joining AIDA, please do let us know. So I know we kind of talked very high level of what we have been doing and what we want to do. I just hope that it painted a picture of what are some of the areas that we can come together to work on and to address some of the challenges that we are facing. So hopefully in the next, today and tomorrow, we can identify what are the strengths of the HCA Asia community and the scientists, and how we can utilize their know-hows and techniques to build further collaborations in the region. Hopefully, we can also find common needs through flagship projects, and through the steering committee, we'll be happy to organize and help to facilitate a lot of these discussions and scientists to formulate these efficient flagship projects going forward. So I just want to remind you again that feel free to join us tomorrow at day 2 breakout session number 3, that we will kind of touch upon these issues in more detail to put ideas into action. So hopefully your participation will be greatly appreciated. So I just want to close by thanks to Shyam and Unyang for a lot of their inputs into this, and Yoshinari Ando and Piero Caninci, who really brought the HCA Asia concept to fruition, and CCI for funding a lot of the projects that I've talked about today. So with that, thank you everyone, and hope to talk to you again later tomorrow.
Thanks, Jay. Very good talk. And we have a question on the Slido from Julia Zhu. For difficult genetic analysis of diseases like ASD, what's the advantage of using Atlas model compared with conventional methods?
Yeah, I mean, let me try to read the question again. Yeah, I think the Atlas model comes in 2 different dimensions. So of course, Atlas, as we would been talking about, as well as Aviv has mentioned, is it provides a vast cellular diversity and the cellular context. So cell-to-cell interactions or spatial contexts, just understanding how neurons or Schwann cells or other particular cells are interacting, and what are their diversity and their interactions and how they communicate. Just having that context, the microenvironment provides a very rich understanding of any disease, where conventionally looking at bulk tissues were averaged or focusing on particular cell type. So hopefully the concept of Atlas will bring in this resolution of cellular diversity and heterogeneity that will answer some of the challenges that facing disease. But the second component, I think, which you're referring to, is genetic analysis. Again, there are attributions of how much genetic diversity can influence the gene expressions. And I think this is a very much ongoing discussion of how we can understand genetic contributions to gene expression. And one of the examples that have been pioneered by GTEx is, how can the genetic variation in non-coding regions, such as in the cis regulatory elements, enhancers or key transcription factor binding sites, have been really understudied in the field of genetic research, largely because they were just very difficult to associate them to a particular disease or gene expression. Many GWAS studies have identified SNPs in the non-coding regions, but people have been focusing on exons or protein coding regions. So this is really an opportunity to understand how we can attribute these non-coding regulatory regions to gene expression, and how that can attribute to the makeup of a cell or the function. And I think we do need genetic diversity or the genetic landscape to understand this. So that's one of the efforts of the IDA project, as I mentioned, is how the genetic diversity attributes to gene expression. Hopefully that can be a jumpstart to many other tissue cell types that's already included in the human cell atlas.
Great. Now we have another question from Hongming Zhang. Might you share your experience on single cell meta-analysis across different races?
Yeah, that's a really good question. We are exploring that as we go as well. How do you define ethnicity, and that is something that we want to stay unbiased as possible. So for example, in the IDA project, when we collect samples from various individuals, we do ask their, ethnic background. Although those are optional to answer, these are kind of personal interpretation, so to speak, of their background. So it is a difficult and somewhat sensitive topic. So one of the ways that we are trying to stay unbiased is to use their genotype information. So that is using just SNPs arrays. We are able to profile their genotypes and correlating that to the gene expression. So yeah, it's an ongoing discussion. I'd be happy to discuss how we can do this more systematically. But at the moment, we are relying more on the genotype profile.
All right. Because of time, we have a final question from Alistair Forrest. How does HCA Asia handle mapping to different genotypes, population-specific genome alignments?
Yeah, so this, I don't know if I could answer that question properly, but yes, obviously different ethnic backgrounds have different genotype profiles as well. As I mentioned in the IDA project, we are using the SNPs arrays, the genotype arrays that are specifically designed for Asian population. So this is something that we are mapping our genotype to. How to broadly apply this concept to different regions around the world needs to be further discussed. So yeah, I don't know if I'm answering this question appropriately to what Alistair is asking here, but we are using a predefined set of SNPs that's catered to Asian populations for genotyping at the moment.
Thanks, Jay. So we have now come to the end of the plenary session for this morning. And after the break, the audience can transit to different concurrent sessions using the Zoom links provided in the meeting manual PDF file. After the concurrent session, you can come back to this room during lunch break for the Flash Talk series. For the concurrent speakers and chairs in this morning, please move to the speaker chair room now to make sure that there is no technical problem. There is a private link emailed to every speaker and chairs, using which you can go directly into the concurrent speaker rooms. However, if you use the meeting menu to log in, you can click raise hand button. Our operators will move you to the audience room from the speaker room as soon as possible. Thanks.
Hi everyone, I am Winfei Jin. I am Associate Professor in Department of Biology, Shaolin University of Science and Technology, Shenzhen. Firstly, I would like to thank the committee for providing the opportunity that I could share my work with you. Today, I will talk an ongoing project in my lab that we are analyzing the classical T-cell subsets using single-cell RNA sequencing. The title of my talk is Reclassification of Classical T-cell subsets and Identification of Novel Cell Type by Single-cell RNA Sequencing. We analyze 6 enriched T-cell subsets, namely CD4 helper, CD4 naive, CD4 memory, CD4 Treg, CD8 CTL, and CD8 naive. The 16 solid single-cell transcriptome from the 60 cell subsets were projected on T-cell plot, forming a single connected entity. Although each T-cell subset was relatively concentrated on a specific region, they are overlapping and local mixing. Those T-cells were grouped into 9 classes. Among the 9 T-cell classes, 6 of which matched predefined T-cell classes, namely CD4 naive, CD8 naive, CD4 memory, CD8 TMI, and 2 CD4 regular T subsets. There are 3 classes which do not match any CD4 or CD8 T cell subsets. With extended reference, one class matches the mucosal-associated invariant T cells. Different from other T cell classes, either belonging to CD4 or CD8 T cell subset, there is one class that contains about half CD4 T cell and half CD8 T cell, named CD8 effective T cell and CD4 TMI. We name this class as single cell T memory. The CD4 memory and CD8 memory in one class provide an excellent model showing that CD4 T cell and CD8 T cell could reprogram and converge into similar phenotypes. Now we check the last class that does not match any predefined T cell subsets. We identified 56 significant high spread genes, including BSD2, SG50, and OAS1 in this cluster. All the enriched geoterms are associated with endoform signaling. We named this cluster as endoform signaling high T cell. After we identified the cell type of each cluster, we demonstrated the relationship between classical T cell subsets and single cell classes by Sankey diagram. Classical CD4 naive, CD8 naive, and CD4 memory cell mainly grouped into their single cell RNA class counterparts, indicating those T cell subsets are relatively homogeneous. While CD4 helper, CD8 CTL, and CD4 Treg were assigned to multiple single cell classes, indicating the highly heterogeneity of those T cell subsets. The T cells were separated into subpopulations by combining the bead enriched T cell subset and the single cell class. Cell subpopulation deriving from the same single cell class showed a similar expression pattern, while cell subpopulation derived from the same bead enriched T cell subset showed a different expression pattern. We enriched single cell interferon high T cell by FACS sorting of BST2 high cell. Single cell RNA and showed that BST2 high T cell is highly heterogeneous. The expression of level of BST2 in interferon high T is significantly higher than that in other cells based on single cell RNA data. We further separated the non-interferon high T cell into BST2 positive and BST2 negative cell based on whether BST2 was detected in single cell RNA data. The expression profile of interferon high T cell is quite quite different from that of BAT2 cell. In the farmer, high T cell is significantly higher expressed in the farmer associated gene than BAT2 T cell. We removed the interfarmer signaling associated gene from transcriptome and reconducted TCNS. The single cell population did not change except interfarmer high T class disappeared and those cells redistributed into the other cell population, concentrating on naive T cell and memory T cell. Interestingly, interfarmer high T classes show up even when we only analyze interfarmer signaling associated gene, further indicating interfarmer signaling associated gene are major contributor for the formation of interfarmer high T regular cells. BAT2 positive cell and BAT2 negative cell are activated by RTCD3 and CD28 co-stimulation for 12 hours. FACS analysis of CD25 showed almost all BAT2 T cells became activated upon co-stimulation, while only a fraction of BAT2 negative cells became activated. My graduates Xiangru and Xuefei are working on this project. I also appreciate the grants supporting my lab. Finally, thank you for your attention.
I'm Wei Vivian Li, and I'm an assistant professor in the Department of Biostatistics and Epidemiology at Rutgers University. In our poster, we would like to introduce a new statistical method for inferring sparse gene co-expression networks from single-cell gene expression data. And this is joint work with Yanzeng Li, a master's student who just graduated from Rutgers in this year, and is supported by NJX and Rutgers School of Public Health. We know that biological systems often involve tens of thousands of genes tightly regulated in complex and dynamic networks, which could change substantially in different tissue types, developmental stages, and cell states. Therefore, elucidating gene interactions in a network manner is crucial for understanding complex biological processes. By identifying abnormal gene interactions and regulations in disease states, it is possible to reveal the biological and biochemical pathways relevant to disease mechanisms and therapeutic targets. In network analysis, genes are represented by nodes, and their relationships are depicted by different types of directed or undirected edges between the nodes. The rapid advances of single-cell RNA-seq technologies have now made it possible to investigate gene networks across individual cells in a cell-type specific manner. And even though exploratory analysis demonstrated the possibilities of constructing functional gene networks across single cells, both technical and biological complications present challenges to the genome-wide inference of gene dependencies from single-cell RNA-seq data. For instance, we calculated the Pearson and Spearman's correlation for a gene expression dataset of 109 immune B cells after normalization and log transformation. There are 886 gene pairs with a similar Pearson correlation coefficient between -0.15 and -0.14, and 1,302 gene pairs with a similar Spearman's correlation between 0.14 and 0.15. But their association varies in a much larger range when we recalculated the correlation coefficients only using the cells in which both genes were detected. This motivated us to develop a new method called SC-Link to infer sparse gene co-expression networks from single-cell gene expression data. To more accurately infer gene co-expression networks that can capture functional gene modules, SC-Link has two key steps. The first step is to calculate a robust co-expression matrix from the gene expression data to accurately represent the co-expression relationships among genes. In this step, SE-Link proposes a robust estimator for measuring gene co-expression strengths while relying on the cells in which both genes are accurately measured with high confidence. In the second step, SE-Link aims to identify a sparse gene network from the co-expression matrix using a panelized and data-adaptive likelihood approach. We use both simulation and real data studies to demonstrate the efficiency of SE-Link and how SE-Link helps us to improve the identification of functional gene modules, regulatory relationships, and co-expression clues about cellular mechanisms that are active in normal and disease processes. First of all, we use the simulated data to evaluate the performance of gene network inference methods given diverse network architectures and experimental settings. We consider two types of network topology, hub-based networks and power law networks. The power law networks assume that the distribution of the node degrees follows a power law, and our comparison results show that SE-Link has the best AUPR and AUROC scores on power law networks, outperforming 5 state-of-the-art methods for gene network reconstruction, regardless of gene numbers and sparsity level of single-cell data. In hub-based networks, where a few nodes have a much higher degree than the rest nodes, we also observe similar comparison results. We then applied SE-Link to tabular MuRIS database, which includes more than 50,000 cells of 59 cell types. Since the true underlying gene networks are unknown for real gene expression data, we investigated the identified edges between genes and known transcriptional factors. And for each cell type, we calculated the number of identified edges connected to known TFs in SE-Link's results and assessed their overlap with TF and target edges discovered in previous ChIP-seq experiments. Our results show that a substantial proportion of the identified edges by SE-Link were also previously discovered in ChIP-seq experiments. In addition, we found that the gene modules in gene networks constructed by SE-Link are enriched with biologically relevant goal terms in a cell type specific manner, and SE-Link can identify gene interactions that would be missed using a classic approach with the Pearson or Spearman's correlation coefficients. The SE-Link can also be applied to study more specific biological questions. For example, we applied it to immune cells in both tumor and matched breast tissue and found that it can help identify disease-associated co-expression changes and co-expression modules. To demonstrate the application of SE-Link and disseminate the research findings in our real data studies, we developed a web application of SE-Link. This application provides an interactive platform for users to subset and visualize the cell type-specific correlation matrices and gene networks constructed by SE-Link. For easy application of SE-Link to additional single-cell gene expression datasets, we also implemented the methods in our package, SE-Link, which is publicly available on GitHub. You're very welcome to try our SE-Link package and web application and let us know if you have any questions or comments. Thank you.
Hello everyone, my name is Du Xiaomi, a doctor from Northwest Agricultural and Forest University. It's a great honor to have the opportunity to share a little bit of our lab work. The topic of my report is PAC7 promotes CD49F positive dairy-got spermatogonia stem cell to maintain self-renewal. This is the background to my experiment. Spermatogenesis is a complex process that originates with spermatogonia stem cells and depends on it. They are localized to the basement membrane of testicular seminephron tubules of male animals. Similar to other tissue-specific stem cells, the number of spermatogonial stem cells is rare. We want to know what his regulatory mechanism is. So we did a single-cell sequence of Daring-Gold testes. The cells partitioning via T-cell analysis identified 12 clusters and obtained differentially expressed genes. We performed unbiased dynamic cell trajectory analysis of pseudo-time order with spermatogonians from a donor gut. Next, we use magnetic activity cell sorting method to purify CD49 positive cells. Why do we select CD49? Because our single-cell sequence data also suggests that it's feasible. To verify whether CD49F positive cells selected have the characteristics of spermatogonial stem cells. The self-renewal-related marker gene GFR1 and PLCF was significantly higher in the CD49F positive cells than the negative cells. This is an RNA sequence. 1,900 differential genes were obtained from the spermatogonial cells of the CD49F positive and negative groups, SRRT1, TRMT1, and so on. These were significantly differentially expressed. This result shows that the differential genes are distributed in 350 different pathways. We screened some of the important genes in spermatogenesis and found that CBTB16, PAX7, LRM28, BMPR1B, FGFR1, FOXO1 were expressed in higher amounts in CD49F positive testicular cell compared to the fibroblast. This is a moderate PAX7 expression in the test tissue. The lower age, the high expression of PAX7, it remained basically unchanged in adulthood. Immunofluorescence staining detects the PAX7 expression of the protein in adult testes of Daring-Gold, which showed only a very few cells were PAX7 positive in the spermatogonial tubules, and most of them were located in the bases of spermatogonial tubules. These results suggest that PAX7 can be used as a marker of spermatogonial stem cells in Daring-Gold. We constructed its overexpression vectors, then transfected into the Daring-Gold spermatogonial stem cell lines. TLZF, OCT4, ID4, and other self-renewal-related genes were upregulated compared with the black vector group. Western blot Asian showed the same traits at the transcriptional level. To summarize, PAX7 promotes CD49F positive Daring-Gold spermatogonial stem cells to maintain self-renewal. Those genes were expressed higher in CD49F positive cells. My deepest gratitude goes first and foremost to Professor Huang, my tutor. I also owe my sincere gratitude to my lab mates. Thanks for your listening.
First of all, we would like to thank HCA Asia for providing this opportunity to share a tool that we recently developed for cell type annotation of single-cell expression data. Currently, unsupervised clustering is the predominant approach for analyzing single-cell transcriptomics data. The analysis is standard. You would expect an annotated cluster map as the outcome, and the outcome depends on what clustering algorithm and what clustering resolution you use. It also depends on how many clusters you are determined to identify. More importantly, cell type assignment is based on users' knowledge. So given this annotated cluster map, you need an expert to go through each cluster, and the expert will assign cell type to each cluster based on expression profile. This process is expensive because you need a lot of experts to do this job for you, and it's also time consuming. So here we present a marker-based cell type annotation method for researchers. Marker takes advantage of the cell marker databases to convert gene expression matrix to cell type score matrix. And each entry in this matrix is a score of one cell for a specific cell type. So Marker can label individual cell a cell type by calling out the maximum value. Independently, Marker also clusters cells into many groups, and we name it as a label 2. We would assume that cells should have the same cell type label in label 1 if they are clustered into the same groups in label 2. So using confusion matrix, Marker maps label 2 to label 1 to get a predictor annotation. In practice, Marker would run multiple sets of clustering to get multiple label 2s, and Marker maps then to label 1 to get different annotations. Finally, Marker uses voting to get the ensembled annotation. The key component of Marker is a good method for converting gene expression matrix to cell type score matrix. Here we found 4 different methods that were previously proposed for doing this job. Marker also requires a good source of cell marker database, and we found PanelDB and cell marker that cover a broader range of cell types in human and mouse across different tissues. We also need benchmark datasets as well as credible datasets so that we can evaluate performance of a marker. So in 8 combinations, we found the annotations using planar score with the markers from PanelDB always have the largest agreement with the author's annotations for all these 6 datasets. So we finally settled down using planar score as the cell type score method and PanelDB as the marker database. We want to use the Marker to help researchers in the lab to annotate a huge single nuclei RNA-seq quickly. So we applied the Marker to this human heart dataset, and you can see Marker strongly agrees with the author's annotation. To give you a quick summary, we demonstrated that Marker works well for a spectrum of single cell data. Marker can quickly annotate a large single cell expression data with reasonable accuracy. If you are interested in Marker, go to Marker's GitHub and find this tutorial to know how to use Marker for your datasets. Finally, I would like to thank people from Bayer and Broad for helping development of Marker. Thank you.
Hello, everyone. This is Wenfei from Zurich. And thanks for the invitation from Guoji and the HCA Asia committee. I'm glad to present my work here, which is about the cellular composition of brown fat. So why would we study fat? First of all, our world is in a weight gain program since the 1970s. Its progression curve is quite steep, but luckily it's counted by years, so we have plenty of time to react. In 2016, 39% of adults are overweight, while 13% are classified as clinical obese. That is highly correlated with several metabolic syndromes like type 2 diabetes and cardiovascular diseases. And this leads to a huge medical care expense that is over 480 billion per year in the United States alone. In order to control the fat, we need to understand its structure and function. So adipocytes are the building blocks of adipose tissue. Traditionally, they are classified as white and brown adipocytes. The white cells are mainly filled with big lipid droplets, which store energy in the form of triglycerols. On the other hand, brown cells are composed of numerous small lipid droplets and mitochondria, which burns fat into heat to keep our body temperature. And the good thing is that upon stimulation, these two cell types are interconvertible. So at room temperature, the brown tissue looks like this. Some of the cells express UCP1, that is the thermogenic protein, and also it's a marker for the activated brown cells. And when the animal is in cold temperature, its brown fat is activated with smaller lipid droplets and higher UCP1 expression. And when the animal was kept at 30 degrees, the thermogenic function of brown fat is not needed, and it actually acquires a white phenotype with a big lipid droplets and less UCP1 expression. So to characterize the cellular heterogeneity and the dynamics of brown adipocytes, we applied single nucleus RNA-seq for these three conditions, and which reveals 10 populations of brown adipocytes. And each condition have their distinct but partially overlapped set of cells. Of particular interest, we found that the red P4 cells are conserved in all three conditions with a distinct marker of CYP2E1 and ALDH1A1. And then we are curious about if this cell type exists in human. So we took several brown fat biopsies in the deep neck area of the patients that undergo thyroid removal surgery. And again, we did single nucleus RNA-seq. And this map is for the whole brown tissue. That is different from the mouse map, which was only based on the adipocytes. And here we can see a lot of T cells and the B cells, macrophages, dendritic cells, and fibroblasts, endothelial cells, and of course we have the adipocytes and the pre-adipocytes. And further, we can subcluster the adipocytes population into 8 groups. And the mouse P4 marker ALDH1A1 is actually expressed in virtually all the 8 groups. And also the CYP2E1 study shows that P4 cells does exist in the human tissue. Then we studied CYP2E1 to localize the P4 cells within the brown tissue. And as we can see, the P4 cells are within the tissue as well as in the peripheral area of the brown tissue, and also within the blood vessel of the brown tissue. And then we would like to know about the function of these cells. And by injecting an adeno-associated virus that express shRNA, we could knock down ALDH1A1 specifically within the brown adipocytes. And this leads to a massive increase of the surface temperature of these animals and suggests a highly active brown fat. And this is also confirmed by a UCP1 study shows the brown fat is really activated upon the knockdown of ALDH1A1. And then using a metabolomic approach in an in vitro system, we identified acetate as the acetate secretion is decreased upon ALDH1A1 knockdown. And acetate can actually inhibit brown adipocytes activity in a dose-dependent manner, and which suggests that ALDH1A1 regulates brown fat activity through acetate signaling. So as a summary, by using single nucleus RNA-seq, we identified a new subgroup of brown adipocytes that can inhibit thermogenesis by secreting acetate. And this study highlights the power of single-cell genomics for the discovery of unknown cell types. I would like to thank all the members of Christian Wolfram Lab and also Abib for their valuable support. And the work will be available online in the next few days. And in case of any questions, please feel free to send me an email. And thank you for your attention.
Hello everyone, my name is Yan, and I'm a PhD student from the Computational and Systems Biology Department at the University of Pittsburgh. I'm gonna give a 5-minute flash talk on mapping vector field of single cells. RNA velocity is a computational technique developed by Mano et al. And was first published in 2018. The idea is to exploit the kinetics of the splicing process and the relationship between the unspliced mRNA and the spliced mRNA, which can both be quantified experimentally with RNA sequencing techniques. It assumes that for cells at steady state, the unspliced and spliced mRNA forms a linear relationship according to the differential equation of the splicing process. With the regression coefficient gamma hat, the RNA velocity is simply U, the quantity of unspliced mRNA, minus gamma hat times S, which is the spliced mRNA quantity. Generally, given a count matrix from RNA-seq, we can uniquely define the state of the cell based on expression levels of its genes. Since for each cell, we can also compute the velocity for each gene, we have a high-dimensional velocity vector in the expression space, indicating the direction where the cell is evolving. What single-cell RNA-seq and RNA velocity provide is essentially a discrete velocity vector field. We aim to learn this vector field from the RNA velocity data, so that we could gain some mechanistic insights which are not readily obtained using conventional data analysis methods. We used an algorithm called sparse VFC for vector field function learning. Basically, it approximates the function in the reproducing kernel Hilbert space as a sum of Gaussian kernels, so that we can minimize the loss function by changing the combination coefficient vector C here. With the continuous vector field function, we can compute its derivatives, which is the so-called Jacobian matrix. So here we have gene X and Y, and there are two self-activating genes which mutually inhibit each other. This forms a toggle switch motif. Their expressional state rates can be modeled with an activating Hill function and an inhibiting Hill function, plus their own degradation terms. Here's the vector field of the two-dimensional system. Here I also show the visualization of two Jacobians. The first one is the derivative of the velocity of gene X with respect to its own expression level. And you can see a clear red band here indicating a positive Jacobian coming from the activating Hill function. The second one is the derivative of the velocity of gene Y with respect to the expression level of gene X. And the blue band amounts to a negative Jacobian coming from the inhibiting Hill function. The blue negative Jacobian region in the first plot comes from the degradation term. So by looking at the sign of the Jacobian of the vector field, we can actually infer the sign of regulation between genes. Here's a Jacobian analysis on a real dataset of pancreatic endocrinogenesis. So the NGN3 gene is known for inducing the differentiation of pancreatic bus cells, which then become the endocrine progenitors. So here is a 2D UMAP embedding of the data. The blue cells are the endocrine progenitors and the orange ones are the pancreatic bus cells. The Jacobian of PAX4, which is a gene involved in the regulation of differentiation process in progenitors, with respect to the expression level of NGN3 is positive, as you can see here. This shows that NGN3 gene initiates pancreas differentiation by potentially activating PAX4. The bifurcation point is where the endocrine progenitors need to take the decision to become either alpha cells here or the beta delta cells. Two genes, PAX4 and ARX, mutually inhibit each other and form this toggle switch motif. The Jacobian analysis indeed show that the corresponding Jacobians are negative in this region. The Jacobian can also be used to compute the acceleration, and we found that it revealed genes that are strongly regulated in different cell types. In the case of the pancreas dataset, we rank genes based on their acceleration values in each cell type, and found that known functional genes often are genes with highest accelerations, as highlighted here. We can also compute divergence by taking the trace of the Jacobian. The divergence is actually a very informative quantity. It measures the degree of convergence or divergence of the vector field at any point. So here Here are the divergence analysis on two experimental datasets. In the case of the pancreatic endocrinogenesis dataset, we found positive divergence at the bifurcation point, and strongly negative divergence at the differentiated cell types. This makes sense since bifurcation points usually have diverging velocity flows, while differentiated cell types are usually the same cell types. In another dataset of EMT transition, we found mesenchymal cells, which are the endpoints of the transition, have predominantly negative divergence. In conclusion, we found that divergence is a good indicator when looking for endpoint cell types. With that, I'd like to thank my advisors, Yvette Bahar and Jianhua Xing at the University of Pittsburgh, and our collaborator Jonathan Weissman and Xiaojie Qiu at MIT. I'd also like to thank the funding agencies at NIH. Thank you all.
Hello, my name is Florian. I'm a postdoc in the PAPCA lab at the Genome Institute of Singapore. Today I'm very happy to tell you a little bit about RCA2, which is a new method for reference-based clustering of single-cell RNA-seq data that is particularly robust against batch effects. So, let's start with reminding you about the default single-cell RNA-seq analysis pipelines we are all using. So we typically start with sequencing and pre-processing, like alignment, for instance, and gene expression quantification. Then we go to clustering, annotation, as well as interpretation and downstream analysis. And this figure really nicely shows that any mistake you do during clustering will have tremendous effects on annotation, on interpretation, downstream analysis, and ultimately on the biological conclusions that we draw from our data. So we should be very certain about the clustering results and we should trust them. And to address this point, many unsupervised clustering methods have been developed, and they are currently the ones that are most prevalently used. And we can split these unsupervised clustering methods into three main categories. We have k-means approaches like SC3 or RACE-ID, we have hierarchical clustering approaches, and we have graph-based methods, which are currently the most prevalent ones. They're used in SURVET and ScanPy, for instance. And they are so prevalent because they can deal very well with large data sets as well as with small memory. So for instance, you can run very big data sets in very low time on a rather small machine, and you don't need very big memory systems for that. However, all these methods, they do suffer from two main caveats, which are shown on this slide. So the first downside is that all these methods tend to give you different clustering results. And this was very nicely shown in a review article by Freitag et al. Furthermore, unsupervised clustering methods are quite prone to batch effects, which means that your sets don't cluster per cell type, but they cluster according to technical variation. So to address these points, one remedy is to use reference-guided clustering methods. And this was shown by our group before in 2017, and also proposed recently in a review article talking about the main challenges in single-cell data science. So in our original reference component analysis approach, which is a reference-guided clustering method, did suffer from limited functionality and scalability. We only provided one reference dataset, and it was rather difficult to integrate RCA, the original version, into existing RNA-seq workflows. And that's why we thought it's time for an upgrade, and we now very thoroughly demonstrate the robustness of the reference-guided clustering according to batch effects. We show that our method is much more scalable. We have introduced many additional reference panels. We also included now mouse panels, for instance. We also offer new ways for downstream analysis. For instance, we offer an integrated way to perform Go enrichment analysis. And it's also now much easier to combine RCA with existing workflows like SURVET, for instance. So the way reference projection works is that we use a reference data set, which is essentially a table of marker genes for different reference cell types and our single cell data set. And then what we do is we simply compute the correlation between the reference set and our query set, which will give us a representation of our single cell data in a cell type space, and this space we will use to perform the actual clustering. And now I will show you that performing clustering in the cell type space is actually very robust to batch effects. And we show this using site-seq data of PPMCs, which was sequenced according to with two different protocols, 10x and ROPSIC. And we derived a gold standard notation for this data using the antibody fraction of the site-seq data, and we then later use silhouette index to look at the separation of the data in terms of sequencing method and cell type. And in this you see on the left-hand side RCA, on the right-hand side you see surrogate, and you can see that the surrogate clustering really separates the data to sequencing protocol, while the RCA clustering does not. And when we at cell types, we see a similar picture. On the left-hand side, we see that RCA actually merges cell types that come from these different sequencing protocols, and on the right-hand side, you see that they actually do form different clusters, although they're actually the same cell type. On a scale of more methods, a wider range of comparisons, we see that RCA2 achieves a very good silhouette index in terms of protocol separation. It's very low. It's almost as good as that of methods that actively correct for batch effects, but the cell type separation is the best using RCA2. So to finish, please have a look at the methods. It's available on our GitHub page. We are working on the preprint. It will be out very soon. Just check our Twitter for that. I'd like to thank all my lab members, and I'd like to thank you for your attention.
Hi, my name is Qingzhu. I'm a graduate student at the University of Pennsylvania. Today, I'd like to share our study on the development of hematopoietic stem cells in the mouse embryo. There are multiple waves of hematopoietic progenitor cell formation at different developmental stages. One of the most important and interesting waves is the last one shown in this graph, where hematopoietic stem cells, or HSCs, form from a specialized endothelial cell called hemogenic endothelium, or HE. Here's a confocal image of mouse embryo at embryonic day 9.5. You can see in the dorsal aorta and villine artery, some endothelial cells marked by the red CD31 are also positive with a green Armstrong GFP expression, which marks hemogenic endothelium. By embryonic day 10, the newly formed hematopoietic cluster cells attach to the endothelium and accumulate in clusters. The clusters will ultimately detach from the endothelium, enter the circulation, and colonize fetal liver, while HSCs will further mature and expand. Because HE and intra-aortic cluster cells are quite rare cell population, we have so far limited knowledge about the exact molecular pathways that regulate endothelial to hematopoietic transition, or EHT. Therefore, we use the single-cell technique to address this issue by specifically capturing the rare cell populations such as hemogenic endothelium and pre-HSCs using known markers based on functional assays. We also collected less purified entire endothelial population to examine the distribution of cells along the EHT trajectory. UMAP of the cells reveal a continuous trajectory of EHT, where we can see the progression of E to HE and IAC cells. Interestingly, we see two streams of FMB2 arterial endothelial cells converge and then give rise to hemogenic endothelium and intra-aortic cluster cells. Separate collection of endothelium from Vidian artery and dorsal aorta suggests Vidian artery only contributes to one stream, whereas dorsal aorta contains cells from both streams. Furthermore, the UMAP of E10.5 entire endothelium shows that before cells become HE, there is an accumulation of cells. This suggests a bottleneck exists during EHT, and we term this intermediate population pre-HE. We perform RNA velocity analysis and found a marked decrease in RNA velocity in pre-HE population. Some of the cells appear to revert back to an earlier transcriptome state, suggesting a differentiation barrier restricting their progression towards hemogenic endothelium. But after cells become hemogenic endothelium, they differentiate smoothly. We notice that RUNX1 is mainly expressed in hemogenic endothelium, but also in about 7% of pre-HE cells. We therefore hypothesize that RUNX1 might help those cells to overcome the bottleneck. To test this, we performed single-cell RNA-seq on RUNX1 wild-type and heterozygous mice. We found dramatically decreased hemogenic endothelium and intra-aortic cluster cells in heterozygous, and increased number of pre-HE cells. This clearly suggests that RUNX1 dosage regulates the cell passage through the bottleneck to become hemogenic endothelium. To understand the epigenetic mechanisms regulating the bottleneck, we perform single-cell ATAC-seq on E10.5 endothelial cells. This UMAP shows a joint embedding of single-cell ATAC and single-cell RNA-seq cells. Differential motif analysis show waves of transcription factor activation during EHT. Specifically, we see consecutive activation of TCF7, SOC17, and GALA2 as cells transition from arterial endothelial cell to pre-HE cells. To investigate which of these factors might regulate RUNX1 expression, we predicted enhancers for RUNX1. Interestingly, besides the known plus 23 enhancer, we found there is a distal enhancer 371 kilobase upstream of RUNX1, which specifically opens up in pre-HE but shows decreased accessibility in later stages. It is also highly conserved among mammals. We think it might be transiently required in pre-HE to induce RUNX1 expression and help cells go through the bottleneck. In summary, using single-cell analysis, we found the differentiation trajectory that underlies EHT, a bottleneck in this trajectory which is regulated by RUNX1 dosage, a distal enhancer which likely activates RUNX1 at the bottleneck. We also found, which I did not show due to time limit, that there are two waves of progenitor formation post-bottleneck, one of which will give rise to pre-HSC, and the other become lymphomyeloid progenitors. Thank you for listening to my talk.
Hi everybody, my name is Tom and I work for the Sydney Cytometry Facility at the University of Sydney, and I'm going to take a couple of minutes just to introduce you to our R package Spectre. The Human Cell Atlas is all about building cellular worlds, taking cells from multiple species, tissues, inflammatory conditions to create the ultimate reference map. A lot of these data sets at the moment are being generated using single-cell sequencing technologies and related assays. But there exists a range of other single-cell technologies that come under the banner of cytometry that allow us to measure fewer features per cell, usually in the order of 40 to 50 protein targets per cell, but it's able to do this on tens to hundreds of millions of cells in individual assays. And so there exists a complementarity between single-cell sequencing technologies that generate huge numbers of features per cell and cytometry, which generates huge data sets with huge numbers of cells. And these two can work really well together in the creation of these cell atlas data sets. Now, the analysis challenges in high-dimensional cytometry data are different, and a lot of these are to do with the sheer volume of cells that are being analyzed in the order of tens to hundreds of millions of cells. So we developed an R package Spectre specifically to deal with these kinds of analysis challenges. The idea here is to provide an end-to-end analysis toolbox, which allows us to go all the way from data pre-processing all the way through to our quantitative and statistical analyses. The reason we're able to we're able to use this on data sets of such size is by using the data table format. This is essentially an extension of R's-based data frame format, but allows for very fast and very efficient operations on large data sets. And so what we're able to do is take all of the expression data for our cells, tens to hundreds of millions of them, combine that with our sample and analysis metadata all in one giant table and perform very fast operations on it. So we've been able to do this on hundreds of millions of cells in individual data sets so far, and using some other tricks with classifiers, we're able to expand this up. The simplicity of this data format also allows us to run this analysis on any form of single cell data, whether that's generated by cytometry technologies, sequencing technologies, or imaging technologies as well, once we've performed cell segmentation. To deal with integrating multiple batches of data sets together, we've borrowed from the cytonorm approach, which is developed by Sophie van Garsen, essentially identifying consistent populations across batches using reference samples and then performing an alignment to bring them into the same feature space. We can do this both to adjust minor batch effects, kind of day-to-day shifts in marker expression, or to integrate much data generated in different experiments or different panels, as well as different technologies. In terms of the actual analysis, we utilize a couple of tools to do our work for us. We use FlowSum for our clustering and UMAP, or one of a number of others, for dimensionality reduction. Essentially, FlowSum will scale very well to very large data sets. We can then subsample what we've clustered, plot this on UMAP to identify the various subsets that we have. We can also utilize a multi-level profiling approach where if we have rare subsets inside our sort of giant data set, we can extract them, profile them independently, reintegrate them into the data set, which gives us kind of a more fine-tuned, subtle analysis, but allows all of that data to become reintegrated into the complete data set. At the end of that process, we obviously want to be able to get some quantitative and statistical answers out of it. And so we've developed a number of tools within Spectre to very rapidly provide us these kinds of answers, which helps a lot with the analysis. To extend this then into the kind of the reference map sort of context that is generated by the Human Cell Atlas, we were inspired by tools such as Surat and Harmony, where we could transfer labels from a reference data set to our data set using classifiers. And so we've worked out a strategy within Spectre to, once we've aligned data from two different batches or experiments or technologies, we're using a k-nearest neighbor classifier to transfer labels from a reference data set to the new data set, which effectively gives us the ability to integrate what we're doing into kind of the reference data sets made by the single cell atlas community. And at the moment, we're utilizing these to create a number of multi-disease, multi-time point atlases in a number of different inflammatory models in mice, which we think will be an excellent complementary data set to many of the human cell atlases that exist so far. So it just leaves me to thank the team at the Sydney Cytometry Facility, as well as our development group, and Giovanna and Felix, who are my co-developers specifically. If anyone is interested in trying out Spectre, the website is here, or you can email me directly. Thanks very much.
Hello, everybody. I would like to share with you today our RICAN Aging Resource Project titled Single Cell Promoter and Chromatin Atlas in Mice to study the effect of the microbiome in aging. So what we're trying to do essentially is to use unique technologies to address unique questions in unique biology, in this case, germ-free mice. So in summary, in our project, what we're doing is collecting many samples from SPF versus germ-free mice in different age and in different organs or multiple tissues. And we are processing this sample for single cell ATAC-seq as well as single cell RNA 5' RNA-seq and single nuclei 5' RNA-seq in order to obtain chromatin landscapes information, as well as defining transcription star sites of genes, which will give us potentially information about alternative promoters and non-coding RNAs. In addition, some of these tissues have been processed for lipidomics, mass spectrometry, by Arita Lab, and the microbiome profiling was done by Ono Sensei. We have actually sampled quite a bit of tissues and time points. We have 4 time points, 9 weeks, 12, 19, and 24 months old. And we have done that in both female and males, and SPF and germ-free. We have 5 biological replicates for each. We have collected single-cell dissociated samples, cryopreserved them for 5' single-cell RNA-seq. We have whole tissue flash-frozen samples ready for single-nucleus RNA-seq, single-cell ATAC-seq, and mass spectrometry. And we have also generated data, samples for imaging. And this is the list of organs and tissues that we have collected for single-cell dissociation and flash-frozen tissues. And right now I'm highlighting the data which we are generating at the moment. I would like to show you an example of tissues that we have data for. The 5' single-cell RNA-seq, and this is for bone marrow tissues. And what we're showing here is a UMAP emphasized on the transcription star site. And the cell annotation was done by using the tabulas muris published. And we use their annotation and transfer them into our data using deep learning. And we are able to separate here the SPF versus the germ-free cells. And here in this UMAP, we are able to visualize the ones that are aged and the ones that are young. Additionally, what we can do with these data is we can both visualize gene expression, like you would with a regular single-cell RNA-seq, as well as identifying the gene promoter or potentially the alternative promoter associated with expression of this gene, which is quite interesting. And also we are able to visualize highly expressed non-coding RNA in this cluster, which correspond to B cell. And what we like to do is to identify and define the cis regulatory that might control regulations or gene expression of gene protein coding genes. And again, here, the example that I'm using is CD79A. And we can see the different elements, cis regulatory elements associated with this gene, and that it is similar for both SPF and germ-free in young. However, if you look at the age trend, you see that the way the cis regulatory change is very different across SPF and germ-free with age. So that's interesting, and that's the kind of thing that we want to, we can do with this data. We are interested in looking into. And associated with this is what are the transcription factors, the key regulons that might explain the changes of cis regulatory elements functions. And this is what we want to do and we can do is to define these regulatory networks and associate this with gene function. And in summary, so what we want to do with this project is to create a single cell atlas to define gene promoter and chromatin landscapes. And with that, we also want to study the effect of the microbiome on aging. And we want to define networks of regulatory elements contributing to aging. And I would like to conclude by acknowledging these amazing people. First and foremost, my advisor, Aki Minoda, a very smart leader and nice person to work with. And I'm really happy and lucky also to be working with all these amazing people who are helping with collections of different tissues. as well as the bioinformaticians, who are helping me analyzing this data. Thank you very much for your time, and bye.
Good afternoon, everyone. We're going to start this very exciting session on COVID-19. First, we're going to have our keynote speaker, Sunny Xie from Peking University. Sunny is the head of the BioPIC and the ICG. And in the past half a year or so, he's been doing amazing work using single-cell technology in producing, neutralizing antibodies against COVID-19. Very impressive work recently published. We're very excited that Sunny will be presenting this part of the work. Welcome, Sunny.
Wow, thank you, Zemin, for the kind introduction. I also would like to thank Guoji for giving me the honor to speak first. The COVID-19 pandemic has proven catastrophic for public health and the world economy. It has been an emotional and stressful time as China and the world was gripped by the spreading virus. As scientists, we all try to make a difference. I'd like to share with you what we have learned and accomplished during this past months. You all know many vaccines are now under clinical trials. Before safe and effective vaccines are widely available, there is a high demand for effective drugs against COVID-19. So here's a list of drugs that have been tried to treat COVID-19. If you could see the last column of summary, the conclusion is that few of them are effective. We certainly do not have an effective cure at this point. So what are the standards for effective drug? It would significantly cut down the proportion of patients with mild to moderate symptoms turning into severe. Second, significantly reduce the damage of patients' lungs and other tissues. And third, quickly cure mild cases, thus shortening the hospital stay. Now, you all know that plasma therapy has proven effective, yet it couldn't be largely applied due to limited supply. One patient needs 1,200 ml of blood from a recovered, from 3 or 4 recovered patients, or convalescent plasma. The effective component of convalescent plasma is the neutralizing antibodies generated by the human immune system. What's neutralization? When an acid comes, a base can be neutralized, can be used to neutralize the acid. When a virus comes, neutralizing antibodies can prevent it from infecting cells. Compared with small molecule drugs, antibody drugs have few side effects. It's very specific. So our goal is to identify the neutralizing antibodies from convalescent patients, make them in vitro through mass production, and then inject them into patients. This is the Y-shaped antibody with the heavy and light chains. SARS-CoV-2 is an RNA virus. This S protein on the surface of the virus is a trimer that has a binding domain, RBD, the red part. Chinese scientists determined the genome sequence of SARS-CoV-2 in less than 2 weeks after the outbreak, then shared it with the whole world, which provided the basis for diagnosis and therapy. Later on, a few groups quickly reported the structure of the S protein and the binding to the ACE receptor, which is on the membrane of human cells, such as lung cells. So neutralizing antibodies bind to the S protein, which would prevent the virus from binding to the human ACE receptor. Here are 3 successful attempts of neutralizing antibodies to treat human diseases. In particular, the US FDA approved the treatment of multi-drug resistant HIV in combination with other antiviral drugs. In the case of Ebola and MERS, effective antibodies, neutralizing antibodies, were also developed, but the epidemics ended before clinical trials got finished. So I particularly want to mention the work of Michelle Nelsonswick at Rockefeller. He first looked at the neutralizing antibodies of blood from different HIV patients using single-cell clonal amplification. Now you see these 4 different patients have different types of antibodies indicated by each color. Now the green ones are not potent ones. The other colors indicate potent antibodies. The number 4 has more type of potent antibodies. So this really highlights the heterogeneity among different individuals and the need for single-cell genomics. Suppose an HIV patient whose blood has high neutralizing potency. How would you know if he or she has a large number of mediocre antibodies or a small number of highly effective ones? So we must determine the distribution. This is obvious for people working in our community, of course. However, the conventional way of single-cell clonal amplification was just too time-consuming. Now, in recent years, many groups have been pushing for high-throughput single B-cell sequencing. So these are the 4 papers in the past 4 years. My own group were not actually doing this at all before the pandemic. The technology was ready, and we put it into medical use for human neutralizing antibody with very limited time under the pressure. So it worked, and it worked very well, of course, through hard work. Our efforts began on the 3rd day after the Chinese New Year. Richard Cao, the only Harvard group member who joined my PKU group, he and I returned to Beijing on the same day. So at the beginning, we work on coronavirus diagnosis. But when we realized that our expertise, single-cell genomics, could really accelerate the search for the effective neutralizing antibodies against COVID-19, we were thrilled. We started reading literature, designing experiments, borrowing reagents and equipment. Everybody was extremely helpful. On February 2nd, we went to Beijing Yuan Hospital, a designated hospital of COVID-19. We met President Jing, who immediately agreed to collaborate by providing blood samples from recovered patients. Bing and Xianghua, 2 researchers of Yuan Hospital, were trained in our lab by Richard and Cheng Yang, who is our sequencing facility manager. Then they 2 returned to their hospital's P3 lab wearing this heavy protective gears. They were supposed to work no more than 5 hours per day, but they always overworked. In the end, they were isolated for almost 3 months. at Peking University, Richard and Cheng Yang have been fighting day and night to collect and analyze sequencing data. This is the cell typing for the convalescent patient's blood by single-cell RNA-seq. You can see different cell type, different types of B cells. So they develop at the beginning from the bone marrow, then grow into grow in the blood. And we were most interested in the memory B cell and the plasma cell. So at the beginning, each B cell undergoes VDJ recombination. As most of you probably know, the work won the Nobel Prize in 1978 by Japanese scientist Tonigawa. The chromosome 14 of the human genome has 40 different sequences of V sequences and 23 different sequences of D and 6 different sequences of J type, like 3 sets of cards. The recombination of VDJ is like drawing one card from each set of VDJ cards, which has the diversity of 40 multiplied by 23 by 6. That's 5,520 in the heavy chain encoded by chromosome 14. So similarly, chromosome 2 has V and J sets, but not D. So that encodes the light chain, which adds more to the diversity of total of 10 to the 7th to 10 to the 8th types of sequences in a human body. Now, in addition to VDJ recombination, point mutations provide more types of sequences. And each sequence results in a different protein with certain molecular shape and interactions, hence varied binding affinities for a specific antigen. Now, what's interesting is that this 10 to the 7th to 10 to the 8th types of cells, each cell has a unique sequence, they exist before an antigen enters the human body. On the membrane of the B cell is the B cell receptor, BCR, similar to the Y-shaped antibody, except they're just bound on the membrane surface. So when antigens such as SARS-CoV-2 comes, binds to the BCR, recognized by the BCR, then what happens is the B cell gets activated and start to divide. Otherwise, they wouldn't divide. And you start to proliferate. And 1 becomes 2, 2 to 4. And then as a consequence, some of them become the plasma cell, which secretes antibodies into the blood. And that they become the neutralizing antibody against the virus. And the rest of them become memory B cells, something we're more interested in, because on the surface of memory B cell are these BCRs. We want to isolate them, the memory B cells, from the blood with their BCRs. So at the beginning, we only used the 10x high throughput VDJ sequencing kit with no functional selection. We sequenced large number of cells which are binned, from 10 recovered patients in order to feed a high throughput sequencer. Unlike Michel's work, we were not looking for Superman. We were looking for super antibodies. The high throughput of the next generation sequencing can do 10 patients together, 10 to the 5th cells at the same time, as you all know. you see this cells, you know, 6 cells have almost identical have exactly identical VDJ sequences, which means they all came from the same parent cell. That is to say, they are enriched and amplified as a result of this activation that I just mentioned. So our assumption was that the higher the enrichment, the more useful this particular sequence is to the immune system, as it is selected by the complicated, you know, evolution system of the immune system. So once we determine the sequence, we of course could use it to synthesize the protein in vitro. We actually asked a company to do it. In doing that, we get the recombinant protein and then test their binding affinity and neutralizing potency. You know, believe it or not, even for the first patient, we were able to find an enriched sequence that resulted in an antibody with neutralizing ability. However, it only accounts for 1%. So if we have 100 sequences, one of them had the neutralizing potency. So we wanted to improve the yield. So what we did was to use this functional selection, attaching the S protein to this magnetic bead and use a magnet to pull this memory B cells out. And that gave us a yield of successful sequences up to 20%. And then we use RBD linked to the magnetic beads, and that further improved the efficiency. So this was our first Cell paper. We reported 58 sequences, along which we found 14, now 20 potent neutralizing antibodies as candidates. And I want to stress here, compared to the other groups at the early stage, the number of cells we went through was at a much, at least 1 order of magnitude. And the high throughput technology really gave us the ability to select better neutralizing antibodies, because this 10 to the 7, 10 to the 8 type are there, and we only get to sequence a small fraction, say 10 to the 5th. So it's really a probability matter. The more we sequence, the higher the throughput, the large chance we have to find a good one. So this is the IC50 curve. That's the neutralizing curve. The horizontal axis is the antibody concentration, and the y-axis shows the percentage of cells that get neutralized. So IC50 is the concentration at which the inhibition reaches 50%, which is 7 picomolar for pseudovirus, 100 picomolar for authentic virus. Similarly, IC80 is at 80%. So IC50 or IC80 indicate the drug dosage. The lower they are, the more effective the antibodies are. The less the dosage. Some of you might know that Trump received 8 grams of antibody made by Regeneron. That is a very high number compared to the normal dosage in cancer therapy. PD-1, PD-L1 is usually under 1 gram per individual. So if there were a more potent neutralizing antibody, patients would receive a lower dosage. Xiaodong Su and Junyu Xiao, 2 structural biologists at Peking University, determined the structure of neutralizing antibodies bound to the S protein using cryo-EM. So the blue bottom is the trimer, the red one is the RBD, and the purple one is the antibody. It is evident that the antibodies binding block S protein binding to the ACE2 receptor. We found most of the potent neutralizing antibodies bind to RBD, not only from the cryo-EM structures, but also from the comparison between the RBD pooling and the S protein pooling down. Neutralizing antibodies have two usages, one stone that kills two birds. First, it is a therapeutic drug. Second, the prophylactic use as a temporary prevention for people like patients, family members, or medical staff. The prophylactic period, as we measured, is around 3 weeks, but can be prolonged to 3 months with modification of the antibody protein. Dr. Qin Chuan's team in China first established the mouse model for humanized ACE2 receptor. So our animal tests were completed in her lab. They infected the mice with SARS-CoV-2. Then after 2 hours, gave our most potent neutralizing antibody 386-2 with a dose of 20 milligram per kilogram. The green one is the therapeutic experiment. The blue one is the negative control for which irrelevant antibody was given. So after 5 days, a high viral load was detected in the lung. This is for the control in blue color. And the viral load was reduced by a factor of 2,500 in the therapeutic experiment. This is a significant drop indicating the effectiveness of the antibody at this particular dosage of 20 milligram per kilogram. If the prophylactic experiment, in this experiment, we gave the 386-2 1 day before the infection, then after 5 days, we detect no virus in the mouse's lung. Now, this shows the body weight change with time. You see both therapeutic and prophylactic group show little change. A slight decrease in weight could be due to the shot, whereas in the negative control, this decline of the body weight. So I'd like to share with you our results from our second cell paper, which is published online. Junyu and Xiaodong have more interesting structured data, and Qin Chuan's lab made the testing on hamsters, which really give us a much clearer sense of regarding the effectiveness of the drug. Then we developed a new cocktails therapy. So these graphs show the potency of the neutralizing antibody. 386-2 is an order or 2 magnitude better than these other antibodies that we found. So what is the structural basis? In this cryo-EM structure, you see here's the S protein trimer, and there are 3 RBDs, 1 up and 2 down. And the structure shows that our 386 368, I'm sorry, -2, binds to all three of them, either up or down. Okay, so this means the stoichiometry of our antibody binding to the S protein is different from the rest of them, especially the one I showed you with the previous structure. Yeah, so this explains from the structural point why our 368-2 is most potent. Now, Qinchuan's team used a new hamster model, which is more advantageous than the mouse model because the hamster is heavier and shows more severe symptoms than the mouse. It can actually, the hamster can die of COVID-19, whereas the mouse only had the light symptom. So under the previous dosage of 20 milligram per kilogram, we look at the effect of the window of dosage, extending from 2 hours to 48 hours with almost no change relative to the negative control. You see the metabolism of a mouse or a hamster is much faster than that of a human, so everything is accelerated. So roughly multiplying by 7, then you can get the number for human. So for human, 48 hours is about 2 weeks. And that corresponds to the time developed into moderate or severe symptom. So the antibody couldn't be suitable for severe cases, as the severe case should not be categorized. It should be categorized to another kind of disease, and that is the immune system were greatly compromised. So the body weight change were confirmed by the pathological results, which are certainly more reliable. We then evaluated the dosage dependence, changing the dosage from 20 milligram per kilogram to 10, 5, and 2 milligram per kilogram. Now, of course, the lower the better, right? Even at 2 milligram or 5 milligram per kilogram, the antibody drug was still effective compared with the negative control. And this was also confirmed by the pathological results. So finally, I want to talk about the pairing of antibodies of non-overlapping epitopes. So this is 368-2 on RBD, and this is the antibody 629 on the RBD. Now, these two epitopes do not overlap, so this is the crystal structure of both antibodies on the same RBD. So the idea was, if the virus undergoes mutation, one antibody might become ineffective, but the chance for both of them undergoing mutation is extremely small. So therefore, the use of a pair could avoid mutation escape. And this was first demonstrated for the case of COVID-19 by Regeneron. Okay, Dr. Yuan Wang's group in China recently evaluated the neutralizing ability of our two antibodies on the reported coronavirus mutation strains. The most commonly observed mutation strain, 614G, luckily didn't have mutation in the RBD region, hence free of drug resistance. But these three strains, L 452R, V4H3A, and F490L actually did have mutation escape. Our 368-2 no longer have neutralizing effects. So luckily, these three strains have very low abundance in nature so far. Now, as expected, if we use a pair of non-overlapping epitopes, the neutralizing power remains the same, and this is the neutralizing curve of the combo. So finally, I want to show you our latest result, a very interesting one. So if we see how the IC50 or IC80 in this case change as a function of time. So we show the value at 2 time points. So you see for each antibody alone, this 593 is actually equivalent to 368-2. It's our naming system, okay? And the other one is 604. That's very similar to 629 I show you. Okay, so these are the IC80 on the 3rd day, and this is on the 5th day. You see, they are greatly reduced with time. Why? This is due to the mutation escape under the evolution pressure of the antibody drug. So if the drug kills the virus, then the virus gets more mutations, not in the region of where the drug, the antibody binds. But what's so great is, if we use this antibody pair, you see the IC80 does not change with time. Okay, so this is a very pleasant result. So the conclusion is the pairing is the way to go. So finally, as a disclosure, I mentioned the potential conflict of interest. My 2 students and I have formed a startup company called Syngolomics with this drug as the first product. Our goal is to use single-cell genomics to develop antibody drugs. And Syngolomics collaborated with Beijing, a big pharma, to conduct global clinical trials of these neutralizing antibodies. And here I have a list of clinical trials of COVID-19 neutralizing antibodies around the world. And Regeneron and Lilly are ahead of the game in terms of the trial dates. And I want to quickly summarize what I have said. High throughput single cell transcriptome and VDJ sequencing speeds up the development of clinical use neutralizing antibodies. We have found over 20 potent neutralizing antibodies from more than 400 recombinant proteins produced by analyzing 8,600 B cells VDJ sequences from about more than 60 convalescent patients. Cryo-EM structures show effective neutralizing antibodies bind to the RBD of spike protein of SARS-CoV-2. The most potent ones binds to all 3 RBD, 1 up and 2 down. A pair of neutralizing antibody with non-overlapping epitopes can effectively avoid mutation escape. Our animal test demonstrated that our potent neutralizing antibodies exhibit high therapeutic and prophylactic efficacy against SARS-CoV-2. I wouldn't be able to tell you this story if I haven't worked with this really talented, hardworking students and colleagues and collaborators. Many of them I got to know only after the pandemic. All these institutions listed here are involved in this work, and I want to thank the support from Beijing Municipal Government, Ministry of Education, Ministry of Science and Technology, and I want to thank you for your attention.
Thank you very much, Sunny, for the inspiring story. Now let's turn to Q&A session. So we have a few questions, and I guess I'm supposed to read off the question to you. So the first question is from Cheyenne, and the question is, if you sequence 1 million B cells from a convalescent patient, how many of them on average would have an IC50 below roughly like 50 nanomolar? So, you know, what's a chance you get such a good antibody?
From one individual, wow. I gave this number 1 million. 10 to the fifth, we get 1, right? Okay, so 1 million will be about 10. But you see, that's only a small fraction, right? So I always get this question. All these people, they went out from the hospital, right? So they had effective antibodies. Why it was so hard to catch them? And that is because we are sequencing a very small fraction of them, right? We're not sequencing 10 to the 7th to 10 to the 8th. If we do, then we find all of them.
If you do 10x genomics, you're able to sequence about 10 to 20 cells per assay. So you're not at the scale yet.
Yeah, so we have, see, we've been, we could do it one person at a time, right? But like I mentioned, I mean, it's heterogeneous. So in the end, we decided we want to do 10 people at a time because we want to get the best one. We don't care the Superman. We only care about the super antibody. So I mean, roughly just like we're sequencing, I know 1 part per 1,000 or 10 to minus 4. But so in that set, we got a few. I mean, that's roughly the number. Yeah.
Okay. The second question came from Callum Parr. The question is, do patients treated with exogenous antibodies or plasma go on to develop a robust endogenous immunity to COVID-19?
No, I don't think so. I mean, the neutralizing antibody, they just, if they're present in the blood, they stop the infection, and then they all have lifetime, they go away, and then the people will go back to normal. Now, in order to, this is not like a vaccine. Vaccine, on the other hand, would create the immune response that would have the memory effect that lasts hopefully forever. But that is really depends on the disease. HIV, nobody have found the vaccine, and so there was there were side effects of ADE, antibody-dependent enhancement. You could have mutation that makes the antibodies previously generated by vaccine become ineffective. And then they're there, they go there to bind the virus, but they don't stop them. They're not strong enough to stop them, and then they got pulled into cells because there's FC receptors. So the ADE could be a problem if there are a lot of antibodies, but they don't do anything. They're not effective. So there was actually a theory, after the Spanish flu, and the first time, less than 1 million people died. It was sort of similar to COVID, but then the second year, 10 times people died, and that one theory was due to ADE, right? So this is if you were to have immunity among a large population. I mean, this could be a potential problem. But if you just take this neutralizing antibody as a preventative drug, this wouldn't happen. But it's more expensive than vaccine, right? So.
Yeah, I know we're running short of time. Just one final question. Maybe you can give a very short answer. The question is like this. So this from somebody. They said, I read from the news that some college student in the U.S. try to get virus and then sell their blood after recovery to cure other patients. Can you comment on such effort if there's no drug or vaccine available?
I don't know.
Okay, I know. It's just they purposely get sick and then they would, sell their blood.
Sounds
apparently, the U.S. have this approach.
No.
But anyhow, this is probably kind of a hard question to answer.
I got to. Sounds pretty dangerous to me. Was this for COVID?
For COVID, yeah. Yeah, especially young people who have a low mortality rate. Yeah. Anyhow, so this is more of an entertaining question. So yeah, thank you very much. Yeah. Okay.
Thank you.
Yeah, thank you very much, Sunny, for the really inspiring presentation. We have to move on to the next speaker. So before we move on to the next speaker, actually, we can take probably maybe 20 seconds break so I can set up the audio visual. So we'll take a slightly short break, maybe less than a minute. We'll be right back. Hello, our next speaker is Professor Zheng Zhang from the Shenzhen Third People's Hospital and School of Medicine. In the past half a year, he's one of the early groups to really start to use a single-cell sequencing technology to study the immune cells in COVID-19 infected patients. He has published a really nice paper in Nature Medicine, and ever since has done additional work. We look forward to his great presentation. Yeah. Okay, go ahead, Dr. Zhang.
Okay. Yeah, and dear friends and dear professors, and it's my great honor to present our recent study here. So my topic is single-cell sequencing reveals the immunohistochemistry in COVID-19 patients. So, and COVID-19 is recognized under the global pandemic. Up to now, there are more than 40 million infections and more than 1 million deaths were caused by the COVID-19. There is no effective vaccines and treatment. So, compared to previously occurred SARS and MERS, the SARS-2 caused more infections, more deaths in more countries and areas. So you also know the host immune responses play an important role in the outcome of the various infections. So as for the COVID-19, most of the people develop into the asymptomatic status, and some of infections progressed into the mild status. Among these mild and moderate status, many of them further developed into the severe, even critical status, and even to death. So, however, we don't know what is determined on the various outcome of the SARS-2 infections. So, my first question is, where is the SARS-2? So, and to answer these questions, we should develop new tools to identify the infected cells, which will aid for better understanding host in various interactions. Although there are several methods to tracing various infected cells in vivo, and in the case of the human clinical samples, these tools are limited, making the passenger infected cells and the viral reservoir cell type hard to detect. So Professor Edo Amit from the Weizmann Institute and I cooperated to develop an unsupervised pipeline for characterization of the viral infections in single-cell RNA-seq. So ViroTracker can map the single-cell RNA-seq data onto a large of the database of known viral genomes. And also ViroTrack can provide annotation of the infected cell type and enables transcriptional sorting of the viral infected cells and versus bystander cells, and even recover the virus induced program and host factors. So up to now, ViroTrack contains the sequence from the nearly 10,000 virus. So then we enrolled 3 mild patients and 6 of the severe patients with COVID-19. And we also enrolled 4 healthy subjects as control. We took the bath as samples to perform the 10x and further do the single-cell RNA-seq and the single-cell T-cell-seq. So mast-cell analysis grouped the mast cells into the myeloid lineage and lymphoid lineage and epicellular lineage, and each lineage was further subdivided into the smallest subsets. We also found there are some key difference in the composition of the bath samples from mild and severe patients. So, and we applied the ViroTrack onto the data generated from the 9 SARS-2 infected patients. And we can see the SARS-2 transcripts were detected in all 6 of the severe 6 samples from the severe patients in variable amounts, ranging from the less than 400 to the more than 50,000 transcripts. By contrast, no viral rates were detected in the 3 mild patients. The coverage analysis further revealed the majority of the viral rates mapped to the 3 ends of the virus segment. So next, we compared the various positive cells and various negative cells, we observed a very strong enrichment of the SARS-2 viral race in the stellate cells and epicellular progenitor cells. And these two cells subsets was known as the cellular targets of the SARS-2 because they express high levels of ACE2 and TMPRSS2. So very interesting, we also observed the enrichment of the SARS-2 race in the SPP1 positive macrophages, and which suggests that the SARS-2 can infect the immune cells or the SPP1 positive macrophages can cytosis viral infected cells or particles. So then we compared the transcriptional expression between the viral positive macrophage and negative macrophages, and we found that as compared to the UMAP negative macrophage, amount of the chemokines such as the CCL7, 8, and the CCL18 was highly expressed by the UMAMI positive macrophages. So, and this section we concluded that first we developed the viral track. This is a computation framework to analyze the host virus infection map. And the viral track can sort infect cells from bystander cells and reveals the virus-induced expressions. And the third, the SARS-2 can infect the epicellular cells and alter the immune landscape in severe patients. So next question is what type of the immune response in the lung ensued after SARS-2 infection. This UMAP finger shows a total of the 32 the clusters according to some typical markers. And in general, these cells could be divided into the macrophages and neutrophils, dendritic cells, NK cells, T cell, B cells, and plasma cells, and epicellular cells. So, and more importantly, we found the macrophage and the neutrophils proportion was increased in the BAF from severe patients as compared to the mild patients. By contrast, the MDC, PDC, and the T cells was largely reduced in the severe patients. So this data shows that there is an unbalanced immune cells in the BAF of the severe patients. So next, we further focus on the macrophages, and we comprehensive analysis the macrophage subset. So according to some typical markers, and the macrophages can be divided into the FCA1 positive, the group 1 macrophages, and the FCA1 low SPP1 positive group 2 macrophages, and the SPP1 positive group 3 macrophages, and FABP4 positive group 4 macrophages. So what is the difference between the functions of these 4 macrophages? So from the table, we can show that the group 1 and the group 2 macrophages are M1-like macrophages, and the group 3 is M2-like macrophages, and the group 4 is similar the allo macrophages. Then we found the FCA1 and the SPP1 positive macrophages were significantly increased in the severe BAF as compared to the mild BAF. By contrast, the FABP4 positive macrophages was reduced in the severe patients as compared to the mild patients. So this data shows that the pro-inflammatory macrophage was largely enriched in the BAF of the severe patients. So finally, we further detected the mRNA expression of the cytokines and the chemokines by BAF. So, and we found the L1 beta, L6, and the TNF alpha was significantly increased in the macrophage from the severe patients, and which was not shown here. And we also found various chemokines CCL2, CCL3, CCL4, and some other chemokines was expressed at higher levels of the in lung macrophages from the severe patients. And also we found the CXCL9, CXCL10, CXCL11, which can attract the T cell and NK cells, was much higher in both mild and severe patients as compared to the healthy subjects. So in addition, we also found detected the cytokine and chemokines in the BAF. So we found the severe patients have much higher levels of the inflammatory cytokines, particularly for L1 beta, L6, L8 in the BAF. And very interesting, there are 3 critical cases. The BAF was persistently detected. So we found that these 3 cases displayed the persistent high levels of cytokines. So what happened in the lung tissue in the COVID-19 patients? Here we show the case. This case is a 66 years old man with pre-existing hypertension. So the clinical conditions of the patient progressed, deteriorated after hospitalization, and he eventually succumbed despite of the many attempts to save his life. So we detected the dynamic the SARS-2, and the dynamic monitoring of the SARS-2 shows the various presence in the BAF of the upper airway samples for more than 23 days. Although the SARS-2 is undetectable 17 days later in multiple samples, we still identified the viral RNA in his lung tissues by qPCR, and viral presence further confirmed by the ensued RNA hybridization. Moreover, viral NP and spike protein in the lung were detected by the immunoblotting. The kernel virus-like particles were also identified by the electron microscopy. So this is very interesting to observe the SARS-2 persistent in the lung, although the upper airway samples remained the virus inactive. So which suggests the viral reservoir and ongoing viral activities in the SARS-2 infected lungs. So more importantly, we also observed obvious lung fibrosis in this patient, as indicated by the cellular threat, muscle staining, and collagen HC. So which suggests the severe COVID-19 can cause the lung fibrosis in this case. So to further explore the potential molecular mechanism underlying the lung fibrosis in the severe COVID-19 patients, so we mapped the receptor ligand pairs among the different cell subsets to construct a cell-to-cell interaction network. So the center of the interaction network in control was of endocellular cells and fibroblasts, so which is likely a requirement for the normal lung homeostasis. By contrast, the macrophage and fibroblasts move to the center in the COVID-19 patient, so which suggests that the macrophage-derived signals might play an important role in the shaping the patient lung microenvironment. So the analysis further shows the CXCL12 highly expressed in the patient fibroblast, which might enhance the increment of the multiple immune cells. And in addition, macrophage produce more alpha beta and TGF beta to moderate the fibroblast functions in this patient. So in it, we found that the macrophage produce more alpha beta and TGF beta, and in it, the patient fibroblast shows the activated phenotype and increased the expression of extracellular matrix such as collagens. So taking together, our data highlights the crosstalk between the macrophage and the fibroblast through the well-known alpha beta and the TGF beta signal pathway as the underlying driving for the lung fibroblast in the severe COVID-19. So for this section, we conclude the single-cell landscape of the bronchial immune cells in COVID-19 patients revealed by the single-cell RNA-seq. And we also observe the pro-inflammatory monocyte-derived macrophage was abundant in the BAF from the severe COVID-19 patients. In our case, we found a persistent viral activity, cytokine storm, and lung fibrosis. So here. So then we analyze the TCR clone type within the T cells from the COVID-19 patients. So these cells, T cells can be divided into the 7 subsets according to these typical markers. So very interesting, we found the clonal expanded CD8 T cells show the aggregative distribution with the transcriptional homogeneity. And while the clonal expanded CD8 T cells from the severe patients show the diffusive distribution with the transcriptional heterogeneity. Then we take the property of this defined highly clonal expanded CD8 T cells in the mild patients. They show that there is a significant increase in the tissue resin markers for this expanded CD8 T cells from the mild patients. So this shows that the highly expanded CD8 T cells may actively participate in the controlling of the SARS-2 infection. So we conclude that CD8 T cells in severe patients BAF was less expanded, more proliferative, and more phenotypical heterogeneous. By contrast, there are a large proportion of the CD8 T cell effectors with tissue resident and highly expanded features were present in the milder patient BAF. So here we propose a model to describe the immune landscape of BAF in SARS-2 infections. So, and as a model, in the mild patients after SARS-2 infection, the lung resident CD8 cells was clonally expanded, which may link to the various clearance. By contrast, in the severe patients after SARS-2 infection, and the effector cells can produce a amount of the chemokines, which further recruit more pro-inflammatory monocyte neutrophils and some other CTLs into the lung, and which leading to the inflammatory injury. So The third question is what's the difference of the immune response between the lung and the peripheral blood in SARS-2 infections? Here we pair the PBMC and BAF and perform the single-cell RNA-seq and the T-cell-seq. We wanted to answer two questions. The first is the difference between the systematic and the local immune response during SARS-2 infections. The second is whether cell migration from the peripheral blood into the lung during the SARS-2 infections. So first, let's say the peripheral blood immune cells. So, and in the peripheral blood, the immune cells could be divided into the T cells, B cells, monocytes, and some other cells. So as compared to the mild patients, the severe patients displayed the increase of the monocytes, especially for the CD14 positive monocytes and the decrease of the T cells and NK cells. Then we focus on the myeloid cells in peripheral blood. We found, so we compared and because the CD14 positive monocytes was largely increased in the severe patients as compared to the mild patients. So we compared the differential expression of the transcripts in CD14 monocytes in the three groups of the subjects. And it is found that the interfer pathway was both increased in the mild and the severe patients as compared to the healthy subjects. But we found the neutrophil activation pathway was briefly enriched in the monocytes from the severe patients, but not mild patients. And especially the S100A8, A9, and A12 was significantly upregulated in the monocytes from the severe patients. So by contrast, the type 5 interfer and antigen processing and presentation pathway was significantly reduced in the monocytes from the severe patients. And for example, the MRC2 molecules and the cytokines. So this data shows the remote the monocytes of the peripheral blood in the severe COVID-19 patients. So we further set up the capillary score and the MRC2 score and found the monocytes from the severe patients displayed the high levels of the capillary score and the low levels of the MHC2 scores. We further confirmed the decreased HLA-DR by peripheral monocytes in severe patients. In addition, this co-signature score was found to positive related plasma inflammation levels and negative with the peripheral T cell counts. So this data shows that the peripheral monocytes displayed the MDIC-like or immature monocytes status, which was involved in the immune paralysis in severe patients. Then we compared the
just 1 minute, Dr. Zhang. Maybe just 1 minute.
Okay, and I will be quick. So here we compared the peripheral and the BAF monocytes, and we found the peripheral monocytes displayed the MDSC-like by contrast in the lung as PD-1 positive inflammatory monocytes. So, and this is the peripheral T cells. And the T cell sharing analysis, there is a peripheral activation of peripheral CD4 T cell response in the COVID-19 patients. And also we found that there is an active recruitment of the peripheral CD8 T cells into the lung in severe patients. So this is concluded for this sections. Okay, and we thank our team and our operators. So thank you for your attention.
Thank you so much. So let's see, we have questions. so far we don't see a lot of questions, but let me ask you a question. I think I previously had a similar kind of discussion with you. So in your different part of the presentation, you talked about the elevated expression of certain cytokines. In some cases, you also mentioned the overexpressions of S100A9 and A10. So such increased expression can be explained in, well, two ways. One is the same cells will somehow turn on their gene expression for chemokines, like CCL8 or some other genes. The second model, though, is not that the cell that turn on the expression, is that the population have a different mixture of different cells. The new cells already have higher expression of those genes. So in that case, you're arguing for not the upregulation, but just recruitment of additional cell types. So in your opinion, which one is more likely?
I think the latter maybe is a major reason. And I think the maybe monocytes from peripheral blood was recruited into the lungs and where and maybe tend to the inflammatory and macrophages. And this macrophage can produce the high levels of the chemokines.
All right. Yeah. Okay. Also, you did mention that S100A9, A10 genes are upregulated, but based on the heat map you showed, it seems like their expression level actually pretty consistent in healthy, mild, and severe symptoms patients. Actually, I don't know. Maybe it's a visualization thing, but I don't know if you recall such results where the S100A9, A10 genes. You did highlight that the genes show upregulation, even though the picture shows fairly consistently high expression.
You mean the peripheral blood, the monocytes can persistent expression the S1?
The macrophages. Right.
Yeah. And so and maybe yeah, and now and we are doing detect continuously to detect these cells, but this data have not shown here.
Okay.
And, yeah.
All right. Yeah, great. Yeah, I think we're running short of time, so I want to thank you again for those for generating really important data sets and those nice findings. Thank you very much.
Okay, thank you.
So, our Dr. Sarah Teckman. Hello, everyone. Our next speaker is our third keynote speaker, Dr. Sarah Teckman from the Sanger Institute. Sarah does not really need introduction to the single cell field. She has been one of the really notable leaders in the single cell field overall, but particularly in the HCA world. And so I really like the subtitle in his presentation, Be Bold, Be Brilliant, Be Kind, that precisely describes who Sarah really is. Sarah, go ahead.
Thank you, Zemin. Thank you for the opportunity to speak here. It's a real pleasure. And as you said, the HCA has kind of been my full-time hobby sort of over the past 5 years or so. And it's really fantastic that the community has generated huge number of suspension cell data, which is what I'm highlighting on this slide, from many, many different organs and many samples from around the world. And so when the COVID-19 pandemic hit us in a really unprecedented way at the beginning of the year, what was good about the situation, about what's otherwise a tragic situation, is that the community kind of pulled together really, really quickly. And in the HCA community, the COVID-19 pandemic sort of catalyzed the coming together of the biological networks, including the lung cell atlas network. And so this is basically a community of people who are interested in airways. And of course, that's one of the major tissues that's affected by COVID-19. Not the only one, but one of the early tissues that the focus was on due to the respiratory distress symptoms. And so what happened was that the lung cell atlas biological network formed. You can find the coordinators for all of the networks on the web, and I'll come back to them later. And what this enabled was also a rapid communication across people focused on this tissue, and then we broadened it out to other tissues. And so the coordinators for lung are Martina Wayne, Pascal Barbry, and Sasha Mishar and Jay Rajagopal. And it was really out of the community response of how we can use, in the first instance, just the healthy reference data from the human cell atlas that we had at the beginning of the year to learn about the mechanisms of the COVID-19 viral infection. And what that led to was basically resources and portals for data sharing that are shown here. And then also the first HCA consortium papers that you can see highlighted in red at the bottom of this slide, which is, our first contributions, and I'm sure there are many more to come, focusing on patient tissues like you heard in the previous talk, to add to our understanding of this, the infection and the pathology that ensues. And so the way we started off as a human cell atlas community to understand the disease was to use that healthy reference data and to pull together everything that had been that was available from the community, both published and unpublished, to learn about the distribution of the viral entry receptors, so ACE2, TMPRSS2, and other proteases, and what's their cell type specific distribution across tissues. And so what happened was that Ward and Sungnak, together with Ni Huang and my group, collected all the data that we had, both from ourselves and other published data, and then reached out to the broader community, together with the Lung Cell Atlas Biological Network and support from everyone, to find out whether there were unpublished data sets for other tissues, such as the eye, the gallbladder, the heart, and so on, where there are unpublished data sets where the authors generously, very generously, rapidly, immediately, basically, on the spot, shared the counts for the cells in those unpublished data sets for the viral entry receptors. And so what I'm going to focus on here is the barrier tissues, because those are the tissues that could be most important in the very early stages of infection, where the virus is meeting essentially the healthy reference data, if you can think about it in that way. And what I'll start with is the nose. And so with Martin Nawan's group, we published nasal biopsy data with bronchial and parenchymal lung data for the airways last year. And when we went back to that data, we found that there are goblet cells, so there are secretory cells in the nose that express the viral entry factors at a very high level. And this could, leads to the hypothesis that the virus may actually be sitting in the upper airway, so not just deep down inside the lung, but up at the top, and that explains some of the high transmissivity of this virus. Equally in the bronchi, lower down, we've got ciliated and club cells that have the necessary receptors. And then deep down the lung, it was known for a long time that there were alveolar type 2 cells that express the receptors already for SARS-CoV-1. There was a paper in 2004 that published this in the Journal of Pathology. So the airways here on the left-hand side have many cells that are susceptible to viral entry that we would predict from the healthy reference data could be infected, and the virus could sit inside. Now, Linda Lako from Manchester generously immediately in early March shared her eye data, and this showed us that superficial conjunctival cells, so in the top of the epithelium of the conjunctiva, express the viral entry receptors at a high level and to a lower extent in the cornea. And what this it suggests is that viral conjunctivitis in could be coming through those superficial conjunctival cells, which also implicates the nasal lacrimal duct in transmission of the infection. And in the gastrointestinal tract, in the intestines, there are enterocytes that express the receptors at high levels. And then in terms of the maternal fetal interface, basically, which is relevant for vertical transmission from mother to fetus, there are cells at on the maternal side, perivascular cells, and on the stromal cells, and then on the fetal side, syncytial trophoblast cells, as well as villus cytotrophoblast cells that have the viral entry receptors. And of course, this vertical transmission is rare, but there have been reported cases, and so this is a potential mechanism for how the virus could jump across the maternal fetal interface. So this healthy reference atlas has given us some predictions and some hypotheses about where the virus could be entering. It provides some explanations about the high transmissivity due to these barrier tissues, the eye, the nose, and so on, that where the virus can be sitting, and also some potential mechanistic insights into the symptoms of the disease, like the anosmia, not being able to smell, and so on. And in this work, we also looked for co-expression patterns and found that the viral entry factors are co-expressed with innate immune factors shown down here in red. And so there's a module basically of that innate antiviral response that's already poised with the cells that have high expression of the viral entry factors. And so I just want to put this up. This is published work, so I'll just go very quickly and say thank you to everybody who contributed data, who are shown here on the right-hand side. And I mentioned some of them, Martina Wen and the other, Pascal Barbry and the other collaborators from the Human Cell Atlas Lung Biological Network, who were key in this collaboration and in interpreting the data. And then Wardon Ni and the other members of my group, who worked really, really hard under a lot of time pressure to deal with huge amounts of data. And all this data is available at covid19cellatlas.org for the healthy donors, which is what I just showed you. And we're now also working with CZI to provide the patient donors from many studies funded by them, but also other studies on this website. So if you'd like to contribute and make your data available through the website, please feel free to write to CV19 CA ingest at sanger.ac.uk there. It's a way of distributing the data. So as we heard in the previous talk, the obvious next steps are to analyze patient samples. So the previous talk was blood and bronchoalveolar lavage. There are also other samples like nasal scrapings and organs from deceased patients that we can study as a community using Cell Atlas technologies to try to learn more about the mechanisms of the disease. So I'll talk briefly about nasal scraping data, and this is a collaboration with Kirsten Meyer in my program and Marko Nikolic in London. And again, Ni and Christoph doing the data analysis. And what I just want to point out here is that from this nasal scraping data of COVID-19 patients, we do see that secretory goblet population that I presented earlier as a prediction of where the virus could sit. Those cells are the ones that have the high viral reads inside the single cell RNA sequencing data. So to confirm the prediction is it actually happens in the patients that the virus enters there. And then in addition, what we find is an inflammatory epithelial cell that has also viral reads. It's a new state that we don't see in healthy reference patients and where we have activation of innate immunity at a very high level. Now I'm going to go from the nose to the mouth and look at exciting data from biopsies from salivary glands, which are shown here, kind of on the inside of the lining of the mouth, and then gingiva, which is here underneath your teeth. And this is from the new human cellulose oral and craniofacial network, which Kevin Bird and Blake Warner, Kevin Bird at University of North Carolina and Blake Warner at NIH have started. And they reached out to us, and again, Ni Huang is the star kind of analyst of the data. What we then did was to look for the different cell populations in the oral cavity. This is the mouse at the top, and it's the human at the bottom. And just so that it's clear. And then what we did was to look for where the viral entry factors are sitting in the in those epithelial cells, and indeed where we can see them is in ductal and acinar epithelial cells. So in salivary gland ducts and the end of the salivary gland, the acini, and they are filiform and fungiform differentiated epithelial cells that express the ACE2 and TMPRSS2, and those are the ones that are suprabasal. So in other words, they're kind of in the lining of the salivary gland where the virus can actually access, not in the basal layer. And that's the prediction from the healthy reference data, and we validate that prediction with single molecule FISH. So this is data from Paula Powers and Blake Warner at NIH, our collaborators who did these staining experiments. And you can see at the top that the little gray and red dots are the viral entry factors. Pancytokeratin is showing those epithelial cells, and we can see those factors both in the ducts and the acini of the minor and parotid salivary glands, so different salivary glands in the mouth. And then if we look at COVID-19 patients to see if that prediction is correct and the virus is entering into the epithelial cells in the salivary glands in the mouth, then in red is the SARS-CoV-2 RNA. And you can see again in the minor salivary gland, the parotid salivary gland, and major salivary gland, both the epithelial cells in the ducts and in the acini harbor those SARS-CoV-2 RNA reads, showing that again, like in the nose, in the mouth, the prediction of which cell subtypes harbor the virus is correct. So I think this is really kind of, maybe it's expected, but it's kind of satisfying that the computational prediction from the data also shows that it's valid, and it helps us to understand the mechanisms of the disease. It helps us to understand that we need masks. Maybe it was understood in Asia for a long time, but it took us some months here to understand we need to protect our nose and our mouth. And it also has implications for diagnostics, because what it means is that saliva is a good or it could be an equally good diagnostic tissue as the nasopharyngeal swabs. And if we study the shed cells in the saliva, then what you can see here with the pink, where the pink brackets are, is that we can see ACE2 in the shed salivary cells, and we can also see SARS-CoV-2 infection in the bottom row. If you look at the red in the bottom row, the red markers, these are shed epithelial cells in the saliva, and we can see that the shed cells in the saliva are from the salivary glands, so they're not from the upper respiratory tract. We can show that the shed cells in the saliva are from the salivary glands mainly, and they're the epithelial cells, the pancytokeratin cells. That's what this PCR, the sorting in PCR shows us. And if we look at stainings of the SARS-CoV-2 RNA and staining of 16S ribosomal RNA from bacteria, then in these cells from the salivary glands, we can see co-infection or co-presence of both the virus and some bacteria, which is interesting. So also, again, these kind of cell atlas mechanistic studies of the cells kind of help us to understand also about what types of diagnostic approaches could be helpful. So I just want to mention in this section again that all this work was possible through intercontinental collaborations, across the whole world. Just like in the previous talk, where there was a nice collaboration across China and Israel and EU, continental Europe, and so on. And in all of this work, this also became possible through these networks of collaborations. And in case you're interested, maybe Aviv has already mentioned it in her presentation earlier in the meeting, but I just want to highlight again that for particular biological, for organs, systems and tissues, if you're interested in particular ones, please join these networks, because they're really powerful networks for collaboration, for exchange of ideas, and for driving forwards the cell atlases of these fields. And the one that I mentioned, so Kevin and Blake and so on, you can reach them through oral@humancellatlas.org. All of these are summarized at humancellatlas.org coordinators. Please join. Please feel free to suggest another network if there's a system that you're interested in that's not covered here. And I also want to just use the opportunity to again invite everybody to join HCA if you haven't signed up as a participant. And it allows you to receive newsletters, to receive discounts from companies, and to be part of the registry for the Human Cell Atlas. So in the last part of this talk, very quickly, I'm going to talk about a couple of other types of tissue samples, blood and organs. So I've talked about the living patient biopsies and tissue, and I will talk very quickly about COVID-19 organs and blood. So in terms of samples of cells from COVID-19 patients, we're generating a lot of single cell data from blood, as you heard in the previous talk, and I'll present later. So almost 4 million cells now, and then much less from tissues. These are data from Boston from deceased COVID-19 patients, so nuclear data. From autopsy tissue, which I'll just mention briefly, what we have available is many different tissues, of course, from these COVID-19 autopsies. And here you have an example from the lung, where we can see very variable loads in different regions of the tissue. So there are some lung regions with low virus, but other lung regions with high virus. So it's you can see these very distinct patterns in the white and in the yellow box of the lung. And from these autopsy samples, there's of course many other tissues, kidney, heart, and so on and so forth. And Omar Baraktar's group, in collaboration with colleagues in London and Newcastle, is looking at many different tissues from the point of view specifically of the vasculature. And so that's a very interesting approach to understanding end-stage disease from these deceased patients. Of course, from the living patients, we can follow kind of all different levels of disease in terms of blood, peripheral tissue, and here in the UK, we have a network that covers Cambridge and Newcastle with a network of collaborators shown here. And this network kind of got together through a real team science approach that includes the clinicians who are gathering the samples, as well as us on the genomics side, and then computational scientists, John Marioni and colleagues, and so on, working together, having lots of Zoom meetings to discuss the study design of the patients. So we have about 130 patients here from different levels of disease, asymptomatic, mild, severe, critical, and so on. And we've profiled them with the total-seq antibodies. And so what that gives us is the distinction between the cells at the mRNA level, also at the protein level, and we can then calculate the changes in cell proportions. And I just summarize very quickly here that we see there's 3 cell DCs and CD8 T cells going down, but then platelets, proliferating monocytes, and CD14 monocytes here going up on the bottom. And if we look at gene expression patterns, then we can associate sort of signatures with clinical severity levels. And so in the plasma cells, there's more stress factors with increasing severity in proliferating monocytes. There's more of these cytochrome oxidases, these enzymes with severity going from left to right. And we can look for gene expression signatures that are associated with worsening clinical score, like these Fc gamma receptor RNase 2 and so on in the proliferating monocytes, where you're going from worse on the left to better on the right. Now, if we drill into the lymphoid compartment, then if we take together the protein and the RNA markers, we can distinguish many different subpopulations, like you heard in the previous talk, different, for instance, CD8 T cell subtypes. And it's the effector memory cells here that I'm pointing to that we see particularly in the COVID-19 patients with clonal expansions. And we do see motifs that are shared across COVID-19 patients in those clonally expanded CD8 effector memory T cells. And these motifs in the beta chain of the TCR are shown here. And we assume that these are to do with the PMHC recognition of the SARS-CoV-2 because they're from clones that are expanded in the COVID-19 patients, and they're shared across several donors. So the number of donors with the motifs is shown on the y-axis, and the log 10 score, the negative log 10 score of the significance is shown on the x-axis. And so all of these motifs are shared across many patients and are highly significant, across many cells from many patients. So it is, we hypothesize that these motifs are T cells that are responding to the PMHC from the SARS-CoV-2 virus. Although we haven't used tetramers to purify them. So this is potentially interesting. So just to summarize here, I've told you about studying using the Healthy Reference Cell Atlas to predict which cell types are susceptible to infection, and then studying nasal scrapings, oral biopsies from COVID-19 patients to show the cells that are actually infected, and then a little bit of work from the UK on blood cohorts and a little bit on the organs of deceased transplant donors. I want to just finish by advertising jobs in my program. So we're looking for new faculty members. If you're interested in joining us, please go to jobs.sanger.ac.uk or just write to me. So this is at all levels. It's not just junior level, also senior level faculty. And I will finish there and say thank you, and I'll take any questions.
Great, thank you very much, Sarah, for the wonderful progress, especially in the COVID-19 space. So let's see, we have a few minutes for questions. Let me start by asking the viral presence in multiple types of cells, because initially people are under the impression that the virus will only infect epithelial cells because they express ACE2 and TMPRSS2. But I think based on I believe one of your talks slides show the presence of viral sequences not in a small number of cell types, actually in a very large number of cell types. You can detect viral sequences. I believe in this morning's talk by Aviv Rega, she also showed there are presence of viral sequences in more than just epithelial cells. So, I mean, what's your take on those? I mean, what are those real infections, or those are bystander infections? And presumably many of those don't really.
From the ACE2, from the viral entry factor patterns, if we just look at the healthy reference, they're not only in epithelial cells. I mentioned also in the decidua those fibroblasts and perivascular cells, the pericytes, if you remember. So in the endometrium, in the pregnant form of the endometrium at the maternal fetal interface, so these are perivascular cells and fibroblasts that have the factors. And in fact, we see them in other tissues, like in fibroblasts and perivascular cells in the heart too. And then I also mentioned, the trophoblasts and so on. So it's not only epithelial cells. I mean, I was emphasizing the epithelial cells, because I was talking about the nose and the mouth, and in those tissues, it's mainly in those cells. But there are also other subtypes, and there are lower levels in the heart and the cardiomyocytes, but the higher levels that we see in our data in the heart are perivascular and fibroblasts.
How about those different immune cell types? I remember we've particularly highlighted the present virus sequences in macrophage cells, which can explain by maybe the kind of interaction. Maybe they're eating some other cell types, but there are probably the presence of other immune cells, and my recollection is that they don't really express those.
No, they don't.
EC2 or TMPRSS. So
no.
The question is.
I mean, there are two possibilities, right? One possibility is there's a different entry pathway, or the other possibility is that it's ingestion, like you said. So it's eating. Yeah.
Okay. Yeah. All right. So there is you also mentioned the co-staining of bacterial signal and the viral signal there. So that's just, I'm trying to figure out what that really means. So do you think somehow the presence of bacterial species there somehow would enable the viral infection? Maybe the human cell types don't necessarily have the entry point for the virus, but somehow the presence of bacteria will somehow enable those. I don't know if you're implying this or not, because you quickly went over those.
No, yes, yeah. I wasn't implying that. So the way I think about it more, because those are cells, I mean, those suprabasal cells in the salivary glands, they do have the viral entry factors. So, I mean, in that sense, mechanistically, it's not surprising that you see the virus there. It's just interesting, that the prediction is correct. I mean, that the bacteria is there. So my interpretation was more that these are damaged cells, and that therefore they have multiple susceptibility. So that was my interpretation, but we don't really know. I just want to show it because we have the stainings.
Also,
what it also says from a diagnostic point of view, is that saliva is a really powerful diagnostic tissue, I think.
Great. Also,
we have more data on that I didn't present.
You did call for maybe participation at HCA. Maybe someone can contribute to samples or effort. Lots of people, would love to join. Except that, right now, various countries, including China, but not necessarily limited to China, there are multiple countries have more stricter rules on the genetic material sharing. I don't know if you have, when you are working with people all over the world from different countries, do you encounter
this is all so this is just the expression matrix, okay? So we don't share any raw data. So it's simply the expression matrix.
Okay.
It's not identifiable per se. It's not identifiable data. So I don't know whether that makes a difference from a legal point of view for you.
Okay, got it. So let me ask a final question. This question from Guoji Guo. He's asking that ACE2 expression in lung is relatively low. Is there any other potential viral receptor?
So I think that the ACE2 expression, even if it's low, is very predictive of viral entry. So that's my opinion. Because what I've seen, for instance, in the salivary glands is expression is very low. We only detect it in a small percentage of cells, but those are the cells where you see the viral RNA. It's those cells. Same in the nose, although that level is higher in the nose. So I think that, yes, single cell RNA sequencing data has a limited sensitivity. So Guoji's right. Sometimes these levels are very low, but from what I've seen, it's very predictive.
Okay.
Okay.
Great. Yeah. So our time is up. I want to thank you again for the wonderful presentation to the AC.
Thank you, Zemin. Thank you, everybody.
Great.
It was a pleasure.
All right.
Thank you.
Okay, good evening, everyone, and good morning, good afternoon, everyone. So it's my great pleasure to chair this plenary session. I'm Xuegong Zhang from Tsinghua University. We have two great speakers for this final session of the day. The first speaker is Professor Zeming Zhang from Peking University. I think it's not necessary to introduce more about Zeming. So I think he is one of the leading scientists in the area of single cell cancer biology. I'd like to say that prior to his joining Peking University, he had spent over 16 years at Genetic Roche, where currently Aviv works at. So now I will give the time to Zeming, and let's welcome Zeming.
Thank you very much, Xuegong, for the introduction. It is my pleasure to present in this very exciting forum. So I will start by going over our previous work on single-cell analysis of tumor-infiltrating immune cells in individual cancer types. And then I will focus on our unpublished work on cross-cancer comparison of tumor-associated T cells and myeloid cells based on the latest single-cell sequencing. As we all know by now, that tumor is a very complex tissue with the tumor microenvironment influencing many tumor properties, including how they respond to different therapies. Inside the microenvironment, we have, or TME, there are many cell types ranging from fibroblasts, endothelial cells, parasites, to different kinds of immune cells. So this is where the immune cells recognize and combat cancer cells. So this is essentially the battlefield for cancer immunotherapy. We have learned in the past years that those tumor-infiltrating immune cells possess special features that are very distinct from those located outside of the tumor. And different tumors exhibit obvious diversity of TME, although it is not really clear what determines the TME diversity. So the complexity of the tumor microenvironment determined that the single-cell technologies are needed to understand TME. Indeed, single-cell RNA sequencing, either SMART2-based or 10x genomics-based technology can be very powerful in multiple different ways, ranging from this rare cell type identification, the tissue composition, changes of cell-cell interaction, the specific marker gene identification, and cell lineage tracing. So before getting to our unpublished work on pan-cancer analysis of tumor-infiltrating immune cells, let me first just quickly review our published work on infiltrating T cells and set up the background for our new projects. So we started by sorting out different types of CD4 and CD8 cells from liver cancer patients and applied the SMART2 technology. We found that tumor-infiltrating T cells possess characteristics that are distinct from T cells from either blood or adjacent normal tissues. Then we use a combined expression TCR-based analysis to show the developmental trajectory or kind of connection between different T cell subtypes. And this is work initially done by Chen Hong and Liang Tao, Xin Yan, and Yuan Yuan. And using the same technologies, we use lung cancer infiltrating T cells to show the similar kind of patterns and identify the additional T cell subtypes, including the tumor-specific and activated Tregs. Further, we show that various T cell targets may possess very distinct single-cell properties, even though they were previously described as the same type of targets. For example, the CD27 shows more general expression across multiple cancer types, but not other better targets, including CD1, or PD1. We went on further to characterize T cell properties in colorectal cancer patients and derive even more detailed classification of T cell subtypes. We found at least that each of the 20 T cell subtypes have distinct properties or preference in tumor, blood, and JSON normal tissues. After we obtained single T-cell data sets from colon, liver, and lung cancer patients using the identical experimental procedures, we were provided with a unique opportunity to compare and contrast T-cell properties across different cancer types. When we examined T-cell composition in their blood samples only, we found that the differences were very minimal. However, when we examined the T-cell composition in the JSON normal tissues, we found that the dramatic differences, indicating that tissue location is the major determinant of the T-cell properties. Now, tumor-infiltrating T-cells seem to harbor an additional layer of distinction on top of those tissue-based properties. So it seems that T-cell properties are shaped by the combination of tissue location and the cancer types. And such T-cell dominance are statistically significant. And for example, the colon cancer and liver cancer had much higher level of both exhausted T cells and activated Tregs than the lung tumors. By contrast, lung tumors contain a much higher level group of cells called ZNF683 positive CD8 cells. This is a group of cells viewed by us as pre-exhausted T cells. The abundance of such pre-exhausted T cells appear to be associated with clinical features such as patients' survival. We found that those lung cancer patients with higher fraction of pre-exhausted T cells versus exhausted T cells had the best prognosis. By contrast, the lung cancer patients with higher fraction of exhausted T cells versus pre-exhausted T cells had the worst prognosis. T cell properties also influence how patients might respond to drug treatment. We all know that among colorectal cancer patients, only the microsatellite instable or MSSI patients were responsive to anti-PD-1 treatment, but not those microsatellite stable or MSS patients. But the underlying T cell differences were not really clear. Now, based on our single cell data, we found that MSS colon cancer patient had a much higher level of Th17 cells than the MSI patients. By contrast, the MSI tumors contain a much higher level of Th1-like cells than MSS. So we call this group of cells Th1-like cells and not Th1 cells because they have higher expression of interferon gamma and CXL13, but their interferon gamma expression seems to be under the control of a unique transcription factor called BHLH-E40. The higher level of Th1-like cells and the interferon gamma production may be important for the anti-PD-1 efficacy. To summarize this published work, the T cells can be classified into multiple subtypes based on the single cell expression data. It seems like the different cancer types contain qualitatively similar but quantitatively different level of T cells. The differences can be summarized in these small tables, okay? And such differences can impact clinical parameters, including how they might respond to cancer immunotherapies. So where do we go from here? So we obviously want to go beyond the initial 3 cancer types that we studied and try to take a much broader view of T cell properties across all cancer types. To construct a pan-cancer single-cell transcriptome atlas for T cells, so we, including Liang Tao, Shijing, and several collaborators, so we downloaded all possible published tumor data sets with single T cells. But those data sets are not enough for those cancer types that are not already published or not adequately represented. We will go there and go to collect additional clinical tumor samples and generate new single cell data. So we aim to obtain data for at least 5 to 10 patients for cancer patients. At the end, we collected data for 316 donors representing 21 major cancer types. after stringent quality filtering, we obtained data for close to 400,000 T cells. Close to half of the data in our data collection were newly generated by us. So we try to obtain both gene expression data and TCR sequences so that we can do the combined analysis. And so such compendium of single cell data were obtained from various single cell sequencing technologies. So we had to really perform proper data integration to make the data more comparable. To achieve data integration, we employed a three-step pipeline. We first performed the Z-score transformation to ensure that all data sets are adequately mixed. Then we grouped similar cells into mini clusters in order to partially overcome the dropout problems and make the data more stable. And finally, we applied the batch effect removal algorithm Harmony to further improve the integration. So as you can see from the UMAP plots of the cells and the marker gene expression, the cells are thoroughly mixed, okay? And the clustering of specific T-cell cluster seem to merge through these steps of data integration, and the clusters seem to be crisper and sharper. And so each of the cluster is called a metacluster because they represent the cluster of the mini cluster of cells. So overall, we obtained 41 metaclusters, and all the metaclusters were shared by at least 80% of all cancer types, indicating that the data are really well mixed. And each of the metaclusters have unique distributions across tumor, adjacent normal, and blood. So it is fairly obvious that some of the groups show tumor specificity, including those terminal exhausted T cells and those activated or 4-1BB positive Tregs here. So there's a lot to talk about, but let's just focus on several specific findings. So exhausted CD8 T cells are of high interest, so let's focus on these cells for a moment. So in fact, we identified not one, but 4 groups of exhausted T cells. They all highly express exhaustion markers, including TOX, TIGIT, CTLA4, and TNFR, SF9. But then there are 2 major groups of exhausted T cells that we label as ZNF, GZMK positive text and terminal text here. And those groups highly express PD1, TIM3, and LAG3. And but in the terminal text, terminal exhausted T cells, there is we see further expression of BATF, the TOX2, the valine, ENPD1, or MYO7A, multiple genes like this. There are 2 additional rare cell types, including OXPHOS text and TCF7 positive text. So the OXPHOS text shows really low activity in different metabolism pathways, including the oxidative phosphorylation pathways, and showing different here. The TCF7 positive text are almost the same as what's known as stem-like cells. They highly express genes related to lymph nodes migration, including the CCR7 and cell. Okay. So next, we combine the gene expression and the TCR data to dissect the trajectory of T cell exhaustion. So first, on the global scale, diffusion map and RNA velocity show that CD8 T cell could develop from naive T cells to either the TAMRA or TEX here. Now focus on the T cell exhaustion. Now based on the gene expression similarity, based on this MONOCO3 results and RNA velocity, we inferred 2 paths from naive T cells from TEX. One path goes through GZMK positive TAMs, and the GZ. Okay, hang on a second. They pass through GZMK positive TEX. And we name this pathway P1, and the other path that goes through the ZNF 683 positive TRM cells. We previously labeled this as pre-exhausted T cells in lung cancer. Okay. So when we examine the TCR sharing results, okay, we again show the really tight connection between the terminal exhausted T cells and GZMK positive text and this group cells, which is ZNF 683 positive cells. So both expression-based analysis and the TCR-based sharing point to the same major pathway towards the terminal exhausted T cells. Okay. So we further stratified the TCR sharing of terminal exhausted T cells by cancer types, and found that the terminal exhausted T cells show high TCR sharing with matter P1, P2, but this pattern seems to be distinct in different cancer types. So in a few cancer types, you see more stronger preference to the P1 pathway, including renal cancer, thyroid cancer, multiple myeloma. But in a few other cancer types, they show other preference, including liver cancer and lung cancer. A few other cancer types show more distinct pathway that involves ISG positive CD8 T cells. So different cancer types may have different kind of paths towards T cell exhaustion. We found that cancer types also exert significant impact on the frequency of T cell populations. Here we show two different examples. Now the median frequency of terminal exhausted T cells range from the highly abundant in the esophageal cancer and the liver cancer and head and neck cancer. But those cancer exhausted T cells are almost barely detectable in multiple myeloma. Okay. Now, if you look at some of the other pre-exhausted T cell groups, you see high abundance in multiple myeloma and ovarian cancer, but very low level in renal cancer and esophageal cancer. So not all cancer types are created equal. Now, based on the frequency of the tumor infiltrating T cells, we defined major immune cell types. One show the high frequency of TEX, but low frequency of TRM, and the other group shows the low frequency of TEX, but high frequency of the TRM. And these two groups can be further divided into 8 subgroups here, from C1 to C8. And cancer types usually show enrichment of one of those immune types, and such T cell-based immune types provides probably new reference to understanding the overall tumor infiltrating T cell properties. Notably, half of the melanoma type tumors were of the type 2, which has high TEX but low 4-1BB positive Tregs, and consistent with their tendency to respond to immune checkpoint blockade. Another example, the C3, are enriched in both thyroid carcinoma and endometrial carcinoma. So this group of cells represent the tumors with low abundance of terminal TEX and high abundance of TRM and ISC positive activating T cells. So we speculate that these T cell groups are the cancer types that might also benefit from immunotherapy. Now, T cell based immune typing also have potential clinical implications. So first, based on the TCGA large cohort data, tumors of the low TEX low and the TRM high immune types tend to have better overall survival in both pan cancer and individual cancer types such as renal cancer and the lung cancer and the liver cancer. Sorry. And second, in the anti-PD-1 treated melanoma, tumors of the TEX low and the TRM high immune types tend to be responders, and tumors of the TEX high and TRM low immune types tend to be non-responders. So to quickly summarize this portion of the talk, and we have developed various, we have used single-cell sequencing to identify and understand unique T cell subtypes and review their dynamic properties, and different cancer types exhibit distinct properties of infiltrating T cells, which may have different clinical implications. So we just spent time talking about T cell within tumor, but of course, within the tumor microenvironment, there are other immune cell types that are equally important. So we really want to expand our analysis to cover additional immune types, in particular, different kind of myeloid cells. And to this end, we have gone back to liver cancer and used a combined SMART-2 and 10x method to cover a lot of cells. And we started including lymph nodes and ascites in addition to liver cancer, so that we can easily capture more cells across more locations. And so this is a quick example to show that one of the immune cell types was fairly interesting. This is LAMP3 positive dendritic cells, which seem to be have a preference in tumor, and such analysis, such cancer types seem to be derived from both the CDC1 and CDC2 based on RNA velocity analysis, and they seem to have higher migration capability. And only this group of cells and not other dendritic cells have higher migration potential, and this is confirmed by experimental validation. And based on, I'm going to skip some of the details. Just want to point out that based on both mitochondrial mutation-based lineage tracing and RNA velocity analysis, we show that such cells would have a tendency to migrate from the tumor to lymph nodes. In fact, if you look at the overall picture, the immune landscape across the multiple different locations are very distinct. But such distinct features are not stable, and there are dynamic features, including the migration of such mature LAMP3 positive disease from tumor to lymph nodes. We also use the migrations of the macrophage from the tumor to ascites. And we also perform the analysis of myeloid cells in colon cancer. In this case, led by Lei and Ziyi. So you use both a combination of SMART-2 and 10x genomics to characterize infiltrating immune cells in colon cancer. And then they identified the 5 groups of macrophage cells here. And it turns out 2 of the group cells have really strong preference in tumor, so we label those as TAM, or tumor-associated macrophage cells. So one is C1QC TAM, the other one is PP1 TAMs here. So we focus on these 2 subsets of TAMs. We found that their expression properties are very distinct. The C1QC TAMs specifically express genes that are enriched in the complement pathways and antigen presentation and phagocytosis. By contrast, the SPP1 TAMs specifically express genes that are enriched in angiogenesis and metastasis. So it is obvious that these are 2 very different types of TAMs. So based on this, it seems like the TAMs might have very different or even opposite roles. One is pro-tumor, the other is anti-tumor. So we have to keep this in mind when we develop strategies of targeting TAMs. In fact, we use the Ranker mouse model to study the effect of the anti-CSF1R antibody, which is expected to deplete TAMs. It turns out that anti-CSF1R antibody would selectively deplete only a subset of the macrophage, so including this group of cells and this group of cells. But this treatment would leave the SPP1 TAM intact. So this is precisely the wrong subset to deplete for the drug treatment. So this explains why this treatment was ineffective in the clinical setting. So I described that from liver cancer analysis, we identified the LAMP3 positive DC, and from colon cancer, we described the finding of 2 distinct subsets of TAMs here with very different roles. The question is whether these different immune cell types are universal across all cancer types, or they are specific to certain cancer types. So to address this question, we again constructed pan-cancer single-cell transcriptome atlas for myeloid cells, led by Shijing and Ziyi in collaboration with our collaborators here. So we downloaded all possible cancer published single-cell data and supplement such data with new cancer types. Then we and so for each of the cancer types, we would analyze them separately and then try to identify all possible major clusters. And for each of the subcluster, then we will analyze and divide those further. So this is one example showing the results of esophageal carcinoma. Then we can repeat this process for all cancer types. We're able to fairly consistently identify all cancer types, including the monocytes, the dendritic cells, the macrophage. Indeed, we're able to identify the LAMP3 positive disease, the C1QC TAMs, and SPP1 TAMs, although their abundance may vary across cancer types. So they are not quite universal, but they are universal to a certain degree. So let's look at the mast cells and the dendritic cells. So this group of cells, I know are running out of time, so let me quickly mention that for some of the cancer types, their presence are fairly consistent. There are a lot of quantitative variation. But some of the cell types, including the CDC2-derived LAMP3 disease, there is a great variation. So in some of the pancreatic cancer, the NPCs, and head and neck liver cancer, there is higher abundance of these cells than other kidney cancers. Now, if you look at the myeloid cells, they show a much greater variation, indicating that the tissue presence show the higher play a more important role in shaping such properties. But more importantly, we observe the C1QC TAMs and SCPP TAMs across all cancer types, even though their relative abundance and relative abundance against other macrophages are very different. So I know Shigong is probably indicating I'm running out of time, so let me just quickly summarize this, that we have used single-cell sequencing to understand the detailed properties of tumor-infiltrating immune cells. They all have distinct properties from those outside of the tumor, and they have dynamic properties, and different cancer types and even different tumors within the tumor types have distinct immune properties. And there's a lot of complications and mysteries. But with the advancement of single-cell sequencing, many of the mysteries are starting to be revealed. So this will first and help us understand the cancer biology as well as developing novel immunotherapies. So this is just an incomplete list of people I want to acknowledge. You should go to our website to see a complete list of contributors. I want to acknowledge our contributors and collaborators and funding sources. Thank you very much.
Thank you very much, Zemin, for the great talk. We do receive quite a few questions, so I will try to read them. And Ram asked a couple of questions. A beautiful talk, Zemin. So what determines this quantitative difference in the TILs between different tumor tissue types? Also was wondering whether you explore metabolic properties of T cell and myeloid cells in different cancer types or tissue types. So those are questions.
So I guess the question, the first reason is why there is such a difference. We know the difference exists. Okay, this is something that I think we're just starting to appreciate. And but what caused such a differences? And it is very clear because if you look at not just tumor, if you look at just different tissues, normal tissues, you already see differences. So that means tissue location is actually one primary factor influencing the presence of certain cell types. TH17, for example, is highly present in colon, regardless of the tumor. Okay, so that tissue location is definitely the important reason. Why is that the case? Why you need the TH17 in colon? Maybe it's related to the need to fight against bacterial. Maybe there are some other reasons in different locations, but we know that's the case. But why would different cancer types influence the presence of T cells in a certain way? I think we are only starting to understand this, and today we don't have enough time to go through all the details, but we did try to correlate the type of mutations with the presence of certain cell types. For example, the presence of exhausted T cells might be frequently associated with certain type of mutations, including those impacting the epigenetic alterations. But we actually need a lot more data so that we can derive more confident results. So, and in terms of metabolisms, I'm not so sure. We haven't exactly examined the metabolic pathways within the tumor locations with the T cells, but we did notice that certain metabolic properties of individual cell types, patients may have an influence. And I forgot the precise results, but certain, like a BMI, the body BMI factor may actually impact certain of the T-cell cell types. So there are a lot of interesting questions that we should ask and try to address in the coming years. So far, I think that we still need more data.
Yeah, another question asked by Alice there. Probably you have answered part of them. That have you looked at T cell across normal tissues? How much of the difference is due to tissue type, and how much is due to the cancer type?
A lot. I mean, a lot. I think tissue type play a major role in shaping the T cell infiltration level and types. So, but to be fair, we only focus more on the adjacent normal tissues. But you can argue maybe adjacent normal is not quite normal. So, so we are also looking at really normal tissue. The normal tissue is a little bit harder to come by. But we are there are interesting data sets produced by other different people, including Goji, for example, and other human cell atlas. I mean, those type of data will be very useful. And so we focus more on the adjacent normal tissues, which is slightly different. We know that part already showed very tissue-specific patterns.
Okay, I would like to add one question to that. That is, have you looked at the same type of tumor, but of different genetic kind of drivers? Is that so the heterogeneity of is that a factor that affects the T cell type?
I want to say yes, except that I really don't have enough data to show this, because we all know, if you take lung cancer as one example, you know, lung cancer, obviously you have EGFR driven, mutation and MET driven, you have, you know, smoker and non-smokers. So we all know for colon cancer, we know MSI and MSS patients for colon cancer show very different patterns. And is that mainly driven by just simply mutation load? I'm not exactly sure, actually. So I really want to have a formal way to address this. The kind of sample we have collected is at a stage where you can maybe starting to point out maybe certain type of mutations may be associated with certain type of T cell types. But honestly, if you control for the cancer type, you control for the mutation type, you have very few samples left that's useful. So based on the data we have, we cannot formally address those, even though I have a suspicion that it's got to be connected. So, but right now, I cannot officially answer this.
Okay, we have time to ask and answer more questions. So Guoji has a question that is, within one tumor, how would sampling region affect the immune cell composition? What would be the best way to separate infiltrating versus circulating tumor cells?
I know Guoji likes to ask tough questions. Of course, this is a question, you know, based on what I have. I cannot really answer, because right now, in the just like early days of tumor sequencing, you take one sample, you hope that this particular part of tumor represents the whole tumor. But we know that it's not the case. So there's definitely kind of sample bias there. But we're hoping that if you sample, if there is certain bias, and if you do within the cancer type analysis, that kind of variation get averaged out. But you know, at least for us, we have not really done an experiment where you take a large tumor, like for example, kidney tumor, which tend to be huge, or liver cancer that tend to be huge. Then you can sample at different locations, and you can see maybe such differences. We haven't done that, and I know different other groups have done this. They do notice differences. I would imagine that going forward with the development of spatial transcriptomics, I mean, such answers can start to be answered. But for us, very often, to be honest, we don't really have the luxury to really sample multiple locations for a given tumor. you get what you get really, but you're hoping that even though this single sampling may have variation, but you're hoping when you have multiple samples, multiple tumor patients, the kind of final emerging answers will be uniform. So that's what we're hoping for, but I'm sure Guoji is right, that if you sample different location, you're going to see diversity and heterogeneity.
Okay, I need to check the technical people whether the next speaker is online, because we have been trying to get him online and it has not been that successful. So we will continue ask questions. There is a bioinformatics question for Zeming. Group similar, so actually grouping similar cells to mini clusters give an advantage over standard pipeline. So I think the question is that true? What is the rationale behind? Have you tried other data integration method?
Yes, we have, but honestly, when we are trying to do the mini cluster, is done more out of necessity, actually. To be honest, when we are studying individual cancer types where we have small number of cells, let's imagine we have 20,000, we have 40,000 cells. That's something that's more manageable. But if you're dealing with like a couple million cells, in this case, for example, for T-cell analysis, we started with over 2 million cells. I mean, most of the analysis method would fail. So out of necessity, we sort of want to group the cells with very similar expression pattern to like a mini cluster. Then when you do the mini cluster, what it does is that in case some of those genes got dropped out, okay, but if you're averaging information in those, you can still recover some of the genes out. So if you do that, in a way, you're partially solving the dropout problem. I'm not sure if this is the best way to solve the dropout problems, but I can partially solve that problems. And also, when you do that, the results are slightly more stable, actually, because you're leaving out the genes, one's individual cells that somehow have a really particular expression patterns. Okay, so those are kind of the ways. It's almost like we have to use this approach for this project because the number of cells are so huge.
Okay, there are other questions. A question from Heather Lee. That do you have data comparing T cells from matched primary and metastatic tumors? This could give insight into tissue of residence as opposed to tumor type.
We haven't finished them, and so this is obviously lots of people. I would imagine lots of people are interested in such question and direction. Now we have done that as well, and so there are all kinds of questions, all kinds of problems with those actually. So we have a project that we are fairly excited about. Maybe in the near future we can share those. But so far, in my mind, we are trying to solve the problem to one level of time. So you have to look at one individual primary cancer types before treatment, for example, to see what are the basic characteristics of those. Because when you look at the metastasized tumor, the question is, if you see features, is it caused by the remote site, or by the metastasis process, or the primary tumor location? So there are a lot of variables. So if you go very far, you can maybe get kind of questions, the kind of answers that may not be obvious. So I think there are multiple layers you want to go to. We do have some data in that regard, and we haven't really finished the study really.
We have another question. Sorry for so many questions. We have another question from Guangshui Jia, who says, thank you for the nice talk. What is the difference between SPP1 and C1QC macrophage comparing to M1, M2 macrophage classification?
Well, yeah, thank you for the answer to the question, but I'm pretty sure he knows the answer because in your own talk, you pretty much described the presence of two subtypes in colon cancer, bladder cancer, I believe. But to directly answer that question, I believe those kind of the classification don't really correspond to the M1, M2 paradigm. And so this is obviously getting very specific, because for people who are studying macrophage, people like to think about, there's an earlier study based on the Murine system, where people would take macrophage, it would induce the cells into a certain way that the one is M1, the other is M2, okay? And we found that our characteristics of the TAM subtypes don't really correspond to M1 and 2. The reason might be twofold. One is that the M1 2 designation was initially derived from the mouse system. Now, the mouse system may not correspond to the human system. Okay, so even though it's widely known, everyone knows about M1, M2, people talk about it all the time. We also try to match up the M1, M2 feature against ours, and the matching is not so great, and possibly related to that, and possibly related to the fact that, which is second dimension, then it's the separation is mostly derived in the in vitro system. How the in vitro system in the mouse system correspond to the clinical sample situation may not be precise, and initially try to fit everything in the M1, M2 paradigm. They don't really fit well, but regardless, in our papers, because many of the reviewers will ask us to do it, so we'll always try to do the comparison, but they don't, there's not a perfect match. So, yeah, so anyhow, that's
yeah, there is more question coming. Another question from Ram. It's do the different TAM subtypes in CRC correlates with the CMS subtype on all their location, such as the ascending or descending colon? That's about the colon cancer.
So hang on a second, maybe the first part is whether those presents are
what do the different TAM subtypes in CRC correlate with CMS subtype or their location?
Is that related to MSI versus MSS? So hang on a second. So okay. So I don't have an answer to that. So I know there is a question about the descending. There is a different parts of the colon, colorectal tissue. I don't remember that we did it. Even if we did, the short answer is I don't know the answer to the question, because if I had done that, if we knew the answer, I would have remembered, and I don't think we have done that. I recall the presence of two cell types are present in almost all colon cancer patients we have examined. So if that's the case, that really means there is not a strong preference. So,
okay. Okay, thanks for the great answer. And I think I see Guoji is online. So Guoji has some question for you. So I will ask him to.
Just to be sure that we don't have the next speaker, right? So I know I'm going way over.
No, just discussion. For the infiltrating versus the circulating immune cells, is there a good way to, distinguish them?
Everyone's hoping that this might be the case. I mean, it's a great question. So everyone's hoping for this finding where somehow in the blood there is a specific cell type or characteristics that is indicative of what's going on inside the tumor. If that's the case, it'll be much easier to take the blood samples and say, hey, this indicates that in your tumor sample, you have these features. Based on our analysis, it doesn't support this hope. Because based on all the things we tested, we always, in most of the studies, we always try to collect blood samples, we collect adjacent normal samples, we collect the tumor samples. Okay, we're hoping, in almost every single study, we see, well, do we see anything in the blood? Okay, that shows that tumor feature is either the high expression, the high presence of exhausted T cells, or some other activated Tregs or whatever. We don't really see that. Maybe it's because our sample amount is small or something, but they're very different. Okay, they're very different. What's going on in the blood is dramatically different from what's going on in a tumor. So at this stage, you ask me this, I would say they don't correlate. Maybe we haven't classified the blood sample in a way that it's so detailed, we understand so much better. Then somehow we found this minor subtypes that somehow reflect what's going on in the tumor, but it's not panning out. So for those people who are relying on the blood samples as a way to predict a tumor microenvironment, this is fairly discouraging, and it doesn't mean it's never going to happen, because our way of understanding immune cell types are evolving, and just because the answer is a no today to me, it doesn't mean in a few years it won't happen. So I don't really know. I would say no at this moment.
Okay. Thank you again, Zemin, for the great talk and great answers, and especially for the extra long time answers. So we plan to have another plenary talk on equity and ethics issues of HCA, but unfortunately, we haven't been able to get Professor Pasa Majuda from India online yet. So after a quick discussion with the organizing team, so we decided to probably it's time for us to conclude today's session. I would like to take this opportunity to thank everyone for being here so late or so early. And I'd like to say that we received almost 1,600 registrations for this year's HCA meeting from 29 countries. And today we have more than 800 active participants joined the plenary research session and parallel sessions today. And so we really had a very fruitful day. And I'd like to take this opportunity to thank all the people. Already this is only the first day, but I'd like to thank all the people, all the technical supporting people from Guoji's group in Zhejiang, and also someone from my group in Beijing. And we have technical supporting groups from Singapore and from Japan and even from abroad. So really, this event cannot happen without all this great support. So with this, let me just double check. So the PESA is not online yet. So we will conclude today's session, and hope everyone have an enjoyable evening or day or whatever. Enjoy your dinner or breakfast or lunch. Thank you again, and see you tomorrow.